Good morning, ma'am. Good morning, ma'am. Rupita, ma'am, you are not audible. Good morning.
Hello.
Yes, yes.
Yeah, it is audible now, sir.
Yes, you can start.
Okay.
Sir has joined.
Good morning, everyone. Warm welcome to all our respected speakers, distinguished faculty members, researchers, scientists, industry professionals, scholars, and participants joining us from India and all around the world. On behalf of Department of Biotechnology and Bioengineering, School of Biosciences and Technology, Galgotias University, in collaboration with the University of Cyberjaya, Malaysia, I extend a very warm welcome to Day 2 of our 5-day International Faculty Development Program on Frontiers in Bioinnovation, Innovations in Bioengineering, Biopharmaceuticals for Healthcare Industry and Sustainability. I hope all of you have enjoyed yesterday's inaugural ceremony and technical sessions. We sincerely appreciate your enthusiastic participation and the engaging discussions that made day one a great success. So today we have another exciting lineup of distinguished experts who will share their knowledge on cutting-edge developments in biomedical engineering, biomaterials, drug delivery, and translational healthcare research. So without further delay, let us begin our first technical session. It is my distinct honor and privilege to introduce our first speaker of day 2, Professor Bikesh Kumar Singh. Professor Singh is a professor in the Department of Biomedical Engineering at National Institute of Technology, Raipur, with research expertise in biomedical signal processing, medical image analysis, artificial intelligence, machine learning, computational neuroscience, and computer-aided diagnosis. He has successfully guided 10 PhD scholars to completion and is currently supervising 7 doctoral researchers in emerging areas of biomedical engineering. He has served as PI and co-PI for 11 sponsored research projects funded by agencies including DST, ACRB, ICMR, ANRF, attracting research funding over 3 crore. Dr. Singh has published more than 150 research papers in reputed SCI Scopus Index journals and international conferences. He has also authored and edited several books and book chapters in the fields of biomedical engineering, artificial intelligence, and healthcare technologies. His academic excellence has been recognized through numerous prestigious honors, including being listed among Stanford University's world top 2% scientists for 3 consecutive years, the IEI Young Engineer Award, Chhattisgarh Young Scientist Award, Marfatia Award, Gauri Memorial Award, and a university gold medal. We are truly honored to have such an eminent researcher and educator with us today. So please join me in extending a warm virtual welcome to Professor Bikesh Kumar Singh. On behalf of Galgotia University and the organizing committee, we sincerely thank you, sir, for accepting our invitation to be a part of this international faculty development program. We are eager to learn from your vast experience and expertise. Sir, we now request you to kindly deliver your expert lecture. The virtual platform is yours, Bikesh, sir.
Sir, you are muted. So please unmute yourself.
Am I audible now?
Yes,
Yes,
sir.
Can you all see my slides?
Yes, sir.
Actually, I'm getting my voice echoed back. Hello.
Yes, sir. Yes, sir.
Actually, I'm getting my voice echoed back.
Actually, sir, you have logged with the two device. In the first device, you have to mute one device, so it will be not echoed. You have to, yes, sir. You have to mute one device, sir. There are two devices by which you
No,
have
I've logged
connected.
using laptop only. Just whether I've logged twice, that I will check.
Yes, sir.
Hello, I'm audible.
Yes, sir. Yes, sir.
Still
You are
I'm
audible,
getting.
but
Okay, you all are also getting some extra noise.
yes, sir. Yes, sir.
Okay. Let me try to. Hello.
Yes, sir. Yes, sir.
I've
You
already
can.
muted one, but let me try to rejoin in that case. That
Okay,
will be
sir.
better.
Sir, you have log out from one device.
Yes,
Now
yes,
it is
yes. Now
perfect.
I think okay.
Yes, sir.
Yes. So I have to share the screen. My screen is
Yes.
not visible.
Yes, sir. Just click on the screen.
Yes. Now is it visible?
Yes, sir. It is visible.
Okay.
Perfect,
So
sir.
very good morning. Okay. So very good morning to all of you. So I'll be talking about some of the work that is related to multimodal breast cancer imaging, which I am doing in my since my PhD actually. That is computer-aided diagnosis system for breast cancer classification using multimodal imaging. So my content I'll have divided my lecture into 4 parts. I'll be talking about background first. Maybe there will be some faculty members or young scholars who may not be having a background of image processing. So I'll talk very briefly little about what is image, what is digital image, and what is image processing. After that, I'll be talking about breast cancer imaging modalities. Then I'll talk about unimodal breast cancer classification systems and multimodal breast cancer classification systems. So basically, when I say image, and then there is other common data which is used in this medical field is signal. So there is a signal and there is an image. So when I say signal, signal is usually a 1D data. So 1D data means, like for example, ECG will look something like this. Okay, and when this ECG is converted into discrete or digital form, it will be in a discrete form. So it will be something like 0, 0, 0.1, and then there will be 0.2, then again it will come to 0, and it will go to minus 0.3, something like that. So it is a 1D signal, which means that we can assume that 1D means either row or a column, not matrix. Okay, so either row or a column. So signal is represented like this. Okay, but when I talk about a digital image, it is represented in form of matrix. Okay, so whenever I say image, either it is an ultrasound image or a MRI image or a mammogram image, any type of image, when it is saved in a computer system, it is generally stored in form of matrix. So this is a 2D data, and sometimes it can be more than 2D also. Okay, so image is a 2D data, signal is a 1D data, and there are two broad fields. One is called as biomedical signal processing, in which I work, and then there is medical image processing, in which I work. Now, when we acquire the image, like for example, so this is the image. So acquiring the image is a different thing. Okay, you are acquiring the image, so that involves instrument like X-ray machine, CT machine, or MRI machine. So acquiring the image is a different aspect, and then after acquiring the image, we have to process the image. we have to process the image. Like that is, we can also call it as post processing. Means once the image is obtained, we call it as post processing. Okay, so I basically work in this post processing area, not in the acquisition area, because that is a different area of research. Like for example, you might have heard terminologies like medical imaging physics, that mainly involves instrumentation, physics, and principles of image acquisition and image reconstruction. But I work in a post processing, like after acquiring the image, we have to handle the image so that its clinical information can be enhanced and it can be utilized properly. Okay, so image is nothing but we can say in a very simple way, we can say that it is a matrix. If it is a gray image, like you can see this image, it is a gray image. So gray image is generally represented by one matrix, and when I talk about a color image, it may be represented by multiple matrices. Okay, so for example, this is a RGB color image. So there will be one matrix for red component, another matrix for green component, and then another matrix for blue component. So color images and 3D images may be represented by multiple matrices. On the other hand, black and white and gray images are represented by single matrices. Every value in the matrix represents some information about the color or the color of that particular position. Okay, so generally if the values are low, it means that it corresponds to darker region. Okay, and when the value is high, it corresponds to the brighter region. Okay, so I'll not go into the deep fundamentals about image processing, just a basic idea, so that like those who have not worked in this area can just get an idea. So image is nothing but a matrix. So we can define image as a 2D function fxy, okay, where x and y are called spatial coordinates. Now what do you mean by spatial coordinates here? This matrix representation includes what? So this matrix is nothing but, so it is something like this. So this is 0th row, 0th column. This is 0th row, first column. Then we have 0th row, second column. So this is called as space. We can call it as a space. And from space, we get a word called as spatial. So in signal, we use time domain. Okay, like any signal, this is represented in time domain, amplitude versus time. But in image, we call it as a spatial domain. Okay, spatial domain means these coordinates 0, 0, 0, 1, 0, 2. These are called spatial coordinates. Okay, these are called spatial coordinates, and every value at this coordinate, so there will be some value here 22, 23, 24. These values are called intensity values or gray values sometimes. Okay, and I just told you that if the value is higher, it is brighter. If the value is lower, it is generally darker. So this is a general idea of how image is perceived by the computer. Okay, as a human beings, what image we see is continuous. It is not digital. Okay, but for computer, every image is a digital image. So it is generally represented in matrix form, and matrix consists of lot of numbers, and these numbers are called gray values or color values. Higher means brighter, lower means dark. Now what happens actually, particularly in medical image processing area, when an image is acquired, the image may consist of lot of noises and artifacts. Okay, so and along with these noises and artifacts, there may be some unwanted things in the image. Like for example, there may be a lot of extra anatomical details which are not currently required. So a lot of extra portions may be there. So that's why we need to do image processing. Okay, we need to do image processing. So when I say digital image processing, every medical image processing problem is a digital image processing problem. So when I say digital image processing, the image is in digital form, and processing means I want to process it so that its quality can be improved, its clinical information can be improved. So that's why that is what is the meaning of processing. Okay, so briefly I will go to the various steps of image processing. So these general steps are followed for all the image processing applications. Okay, so for example, we have a problem domain. That is, either it can be a medical image, just keep my phone with me in case I get automatically logged out. Please call me. Okay. Just because I'm using Institute Internet, so if by default, if I get logged out, please call me. So once we have a problem domain, I may work in breast cancer imaging, I may work on any other application, not only medical application, any other application. The first step is to acquire the image. After acquiring the image, we have to improve the quality of the image. So we acquire the image and then we have to improve the quality of the image. So that area is called image enhancement. It is also called as image restoration. Okay. In enhancement, what we do, we simply try to increase or decrease the brightness of the image so that some details can be enhanced. Okay, it is a very subjective approach. This enhancement is a very subjective approach. Subjective approach means some technique whatever technique I apply to improve the quality, some people may say that this is better, some may say that this is better. So it is a subjective approach. Like sometimes we apply filters on our filters in our photographs or our selfies. We make it little bright, little dark, and we try to get the best which we like. That is called as enhancement. On the other hand, restoration means the noise or the artifact in the image is standard noise. Okay, so if it is a standard noise, we'll apply some standard technique to improve the quality or to remove the noise. So this is called as restoration. Okay, like there are some standard noises in images, like for example salt and pepper noise, Gaussian noise. So for that we have to use some standard technique. It is not hit and trial. Enhancement is a hit and trial method. We increase the brightness little bit, then we decrease the brightness little bit. So whatever is better we select. But restoration is an objective process. Restoration is a objective process, which means that the technique is fixed here. This is the noise. So for a then we have another field in medical image processing, which is called morphological processing. What does it mean basically? When we try to analyze the morphology, when we try to analyze the structure, we call it as morphological image processing. Okay. Then we have segmentation. In segmentation, if I have an image and I want to separate a particular area, maybe this area is a tumor or the region of interest. So we try to extract that area and remove the unwanted areas, because it will unnecessarily take a lot of computational resources if I take a bigger sized image. Okay, so some unwanted parts can be removed and only the region in which I am interested can be extracted and taken further. That is called as segmentation. Like in this particular image, there is a shadow here. So that shadow should be eliminated. So this is a general concept. But in medical image processing, there may be many areas which are unwanted due to imaging. So we extract only the region of interest. That is called as segmentation. Like if there is an image and there is a tumor, so I only want to extract the region enclosing tumor and remove other things. This is called as segmentation. After that, we have object recognition, and then there is something called as representation and description. So this part actually, we can say, actually image processing ends here, or rather I should say it ends here. Image processing ends here. Okay, this is the end of processing. After processing the image, after improving the quality of the image, after extracting the region in which I am interested, the next part is to extract some patterns from the image. Okay, so that is then what comes into picture is feature extraction, feature selection, machine learning, deep learning. These things come into picture. Okay, so like for example, I have an image, okay, and there is a tumor. So I extracted the region of interest. So now after this, I get a small image with a tumor. Now from this image, I want to extract area of the tumor, perimeter of the tumor, height by width ratio of the tumor. So if I extract these things, then these are called features which are extracted from the tumor. Okay, so this is called as pattern identification, and sometimes it is called as object recognition. Why object recognition? Because from the extracted features, whatever features we extract, like area, perimeter, whatever, so from the extracted features, I am trying to differentiate whether this tumor is a normal one, or it may be cancerous, it may be non-cancerous, whatever. Okay, so this area is called object recognition. And sometimes there is another field called as representation and description. So for example, this is the tumor area. So I may represent that tumor area with the help of some specific, like for example, this representation which you see here is called chain codes. Okay, so I have just fixed some values like this. Okay, like if I move in a upward direction, I'll use 1. If I go in a right direction, I use so for this figure it is 0 for this one. If I come down, it is 3. If I go left, it is 2. So I use this type of nomenclature, and then I represent the tumor with this nomenclature. Okay. So for example, this is the tumor. Just see this figure. This is the tumor. I start from here, this point. I start from here. So now I go right. So the code will be 0. Okay. The code will be 0. If I again go right, it will be 0. I come down, it will be 3. 3, 3, 3, 3. Then again 3. Then I go left. So the code will be 2. And so on. I represent the tumor area with the help of some type of coding. So this is called as chain code. Ultimately, I can say that if the chain code is very long, the perimeter of the tumor is quite higher. So that gives me the idea of spread of the tumor, something like that. So I just wanted to, by this slide, I just wanted to explain the general idea of image processing. Okay. And what are the different steps that are generally used in image processing. I also want to mention that these steps are not always required. Okay, sometimes I will do only segmentation. Sometimes I will do only object recognition. Sometimes I may use enhancement. I may not use enhancement, because nowadays these deep learning techniques, they take care of a lot of things. I may do feature extraction. I may not do feature extraction. And then there are some other fields in image processing which are called image compression, color image processing. I'll skip that. So with this, I'll directly come to breast cancer imaging. Okay. So I'll not discuss about cancer and its biological aspects. Okay. Everybody knows it, that cancer is uncontrolled growth of cells. And particularly in females, one of the common cancers is breast cancer. Okay. So other than cervical, this is one of the very common cancers. And the good thing about breast cancer is that if it is detected at early stages, it can be cured. Okay. It can be treated properly. So whenever any woman in India, if she feels any symptoms, any type of pain in the breast or any other symptom, when the patient goes to the doctor, the first thing which doctor will do is physical examination. The first thing which doctor will do is, okay, so I have covered the first part here. Okay, this was the background. Up to this thing, it was a background of what is image and what are the general steps in image processing. Now I'm going to breast cancer imaging modalities, because we are going to apply this image processing concepts into breast cancer area. Okay, this is the second part of my presentation. So whenever a patient goes to the doctor, the first thing which is done is physical examination. So the doctor generally tries to analyze the breast physically and try to analyze that whether there is some lump which can be felt manually. Okay. And after that, the first thing which is done is mammography. So mammography is an X-ray based imaging technique, most commonly used breast cancer screening technique, and it is very widely used in breast cancer screening. Okay, so particularly in foreign countries, it is generally said that after 40 years, women should go yearly, or at least once in a twice year, they should go for mammography. Okay, because mammography can detect very minute signs of cancer in early stages. Okay, so the first thing which a doctor recommends is to go for mammography. And mammography is X-ray based imaging technique. Okay, so what is done in mammography basically? So mammography is X-ray of breast. It is a screening tool. So very important thing should be noted here. It is a screening tool. It is not a diagnostic tool. It is not a confirmatory tool. Okay, it is just a screening tool. And if some abnormality, and it uses a low dose, so basically what happens, the process is such that the breast is pressed and made as flat as possible, and after that the imaging is taken. Okay, so this is a general way of doing the imaging. So what happens in this is that if there are some minute calcium depositions, okay, you can see some minute calcium depositions in breast. This is called as calcification. These are called calcifications. Okay, so these can be detected by mammograms. It cannot be detected by any other imaging modalities, particularly not by ultrasound or any other imaging modalities. So that is the best. Like that's why mammograms are so popular, because it can detect these early signs of cancer also. Okay, like this is the original image and this is a little enhanced image using image processing. So this image is little more clear compared to the first one. Okay. So basically this mammogram is the first choice, but what happens that there is a problem with this mammogram. There are some limitations of this mammograms. What are those? That mammography has the limitations in detecting cancers in dense breast. So it is not suitable for young patients, maybe patients with of age less than 30 years. It may not be suitable because the younger women, the breasts of the younger women they are very dense and they are full of milk glands, and therefore detection of this calcifications detection of these calcifications in dense breast is very difficult. So it may not be suitable for uh, females less than younger females, I would say, in general. Okay. Then second is that it is very painful and it involves X-rays. Uh, so there is exposure to radiation. So these are some limitations of mammography. Okay. So what is the second choice? The second choice is ultrasound. The second choice is ultrasound, which is based on sound waves. Okay. And there are a lot of mammogram is costly also. The instrument is also costly, and the imaging technique is also costly. It is not very common, but ultrasound is very common. Ultrasound is very common. Okay. So this is the second choice. After mammogram, the second choice is ultrasound. Sometimes even what happens, mammogram is done. If mammogram can detect calcifications, but there may be some tumors also, there may be some lumps also. So that if they are not clear in mammogram, the, the recommendation is ultrasound. So sometimes even both are used, because ultrasonography is not harmful. It does not involve any radiation. It is not a painful process. It is a low cost process. So a lot of advantages are there with ultrasound. So sometimes this mammography is used in together with ultrasound, or vice versa. Ultrasound is used together with mammography. And if machine is not there in hospital, ultrasound is very common. It is always there. So ultrasound is used for screening. And maybe for further analysis, MRI is also used, but I'll not go into MRI because these are the two most commonly used. Okay. Further, for staging purpose or for analyzing the spread, MRI, PET scan, lot of things are there, but the screening tools are mammography and ultrasound. Sometimes mammography is the first preference, but it has been observed that mammography, when used together with ultrasonography, they give better results. Okay. After screening, if some abnormality is observed, the next step is histopathology, where some tissue from the breast is taken out and analyzed in the laboratory, and that is called as histopathology analysis. That gives the final confirmation that whether the tissue has some malignancy, that is cancer, or not, or whether it is a benign one, or it may be some infection or something else. Benign means it is not harmful. There may be some lump, but it is not harmful. There may be cyst or something. Okay, this is the general process of breast cancer screening. As I told you that this is one part, acquiring the image. So once we get the image, we are working after that, not the pre part, the post part. Okay. So I think this I have discussed, and I have already told you that ultrasonography has a lot of advantages, but it has also some disadvantages, like ultrasound is comparatively noisy compared to mammograms. Okay, because they inherently contain a noise called noise, and they also shadowing effects. Like in this particular image, you see a lot of white and black dots. They are called speckle noise. And in this image, this particular image, you see that there is shadowing. So this shadowing sometimes may misguide the clinician. It may appear as a cyst or a tumor, but it is not actually that. It is just an artifact. Okay. So finally, we have just discussed about imaging modalities, that is mammography and ultrasound. So the first step is physical examination, second step is mammogram, third is ultrasound, sometimes all three combined with each other. So for all this, we need the experienced radiologist, and sometimes even experienced radiologists have difficulties to correctly diagnose the breast cancer, because experience always matters, how practice always matters. So young professionals may not be as accurate as those which have experience of 10 years, 15 years, 20 years. Okay. And also there are some other issues. Okay. So this I have already discussed, that we use mammography followed by biopsy, or we do mammography followed by ultrasound followed by biopsy. Sometimes ultrasound alone can be used. Others are MRI, breast elastography, and a lot of other tools are there. Okay. So ultimately, I just told you that there are some drawbacks in the normal procedure. So normal procedure is what? Physical examination, mammogram. Mammogram cannot detect lumps in younger patients. Ultrasound has lot of noise. So what to do? So there is a solution called as computer-aided diagnosis. What researchers started doing? Okay, let's take help of computers. Let's take help of AI tools, machine learning tools, deep learning tools to further improve the performance, diagnostic performance. Okay, to further improve the diagnostic performance. Okay, so I'll skip this particular slide. So that gave emergence to something called as Acha. Another important thing is that the biopsy which we were just discussing, along with biopsy, lot of other pathological tests are required. Okay, so there is a financial burden to the patient also, and sometimes biopsies are done multiple times. Biopsy means the tissue that is taken out, histopathological analysis. Okay, so sometimes that has to be done multiple times to confirm the diagnosis. So these issues are there. So the solution is computer-aided diagnosis. So why we need computer-aided diagnosis? Early detection is one of the reasons. Okay, and it can improve the diagnostic performance. The point here is that we don't want to replace the radiologist. We want to improve the performance of the radiologist. Okay, so a radiologist without computer or AI tool and radiologist with AI tools will result in a better interpretation, will result in a better clinical diagnostic. Large volume of data can be handled by computers. It will reduce radiologists' workload. It will improve consistency and objectivity. So why consistency will improve? Because sometimes what happens, a radiologist sees the image, will have some opinion. Another radiologist can have a different opinion. So opinion of one clinician can be different from other clinician. So there is no consistency. But when computer interprets the things, there may be consistency. Okay, and objectivity is there. There is subjectivity is reduced and objectivity is improved. Also, it can handle difficult cases. It can support in screening programs. Okay, even if expert radiologist is not there, a young radiologist is there with AI tools. So screening, it can be mass screening can be done in rural areas and remote areas, and it can be cost effective in a long run. Why cost effective? Because it will be like radiologist workload will be reduced, so it will be able to screen more images in less time. Okay, so for long term, in a long term way, it can be cost effective also, because there may be less number of biopsies if AI tools and these can detect tumors in early stages. Maybe number of biopsies will be reduced, because biopsy has to be done for confirmation. But the point is that sometimes multiple times biopsy is required. Okay, so number of biopsies can also be reduced if CAD systems are used. Okay, now we come to the basic layout of CAD system. So these figures, these two figures, I have used AI tools to get it done. So basic layout of CAD system, the first step is data acquisition. So it can be mammogram or ultrasound or MRI, any imaging technique we want to work. Now we know that every imaging technique will have some advantages, some disadvantages. So based on the feasibility, based on the availability, based on the financial considerations, some imaging technique is data acquisition, imaging modality is selected. Then we do pre-processing to remove noises and artifacts. Then we go for feature extraction. The features can be based on shape, like we discussed earlier, area, perimeter, something like that. It can be based on texture. So texture is basically like if all the values in the gray in the matrix are same, we can call it as uniform texture. If the value change very abruptly, we can call it as non-uniform texture. So we can extract features related to texture also. We can extract statistical features like mean, median, something like that, kurtosis, skewness. So a lot of features are there. We can extract. There are thousands of techniques for feature extraction. Then we go for feature normalization. Okay, feature normalization is a process in which all the features that are extracted are brought in a one particular range, maybe 0 to 1. Because what happens, say for example, if I am extracting feature 1, its range is 0 to 10. If I extract feature 2, its range is 100 to 1000. Now what happens if higher valued features go to the ML model? ML model may assume that these features are more important because they have very high value. Okay, but that should not happen. Every features should get equal importance. So for that, we normalize all the features in one particular range, maybe 0 to 1 or any other particular range. Then there are different normalization technique like min, max, z score. I will discuss that. After extracting the features, we go for feature selection. So what is feature selection basically? Suppose I extract thousands of features. But all features may not be important. Some features may be redundant. Some features may convey the same information. Some features may not be able to discriminate between benign and malignant tumors. Okay. So we have to eliminate those redundant and unuseful features. So this is called feature selection. So there are different methods like filter method, wrapper method, embedded method. So in filter method, what we do, we use some criteria. So I'll not go into the technical depth, but for example, information gain is one criteria. Then Gini index, you might all have heard Gini index. So based on some criteria, we rank all the features and then we take top 10, top 20, something like that. That is called as filter method. Filter method is a ranking approach. We rank all the features on the basis of some criteria and then we take top 10, top 20. In wrapper method, we don't do that. In wrapper method, we try to find out the best subset of features. So if there are 1000 features, I want to determine which features are best. We want to determine which features are best. Okay. Like for example, some features when grouped together may work better. So that is called as wrapper method. Like for example, I'll just give one example. I take feature 1, I calculate the accuracy, then I combine F2 with F1. If accuracy improves, F2 will be kept, otherwise F2 will be eliminated. Then I combine F3, accuracy decreases, eliminate. So in this way, we try to find the best group of features which gives me the best accuracy. This is called as wrapper. And in embedded method, this feature selection part is embedded in the AI model itself. Like in deep learning, particularly in CNN, we don't do this feature selection separately. CNN is designed in such a way so that it automatically extracts features and takes only good features forward. So that is called as embedded methods, in which feature selection is embedded within the AI model itself. Okay. So we had suppose 100 features here. So feature selection may give me suppose 20 features. And these 20 features are then taken to AI model, machine learning or deep learning model. So it can be support vector machine, artificial neural network. There are so many deep learning, LSTM, CNN, transfer learning. So many techniques are there. And this AI model should predict that whether the image that was given contains a tumor. And if it contains a tumor, whether it is malignant or whether it is benign. And there may be sometimes multiple classes also, not only malignant and benign. There may be multiple classes also. Okay. So in this way, we move forward. Now, I'll just skip few slides because we have a limited time. I'll just discuss some of the studies which we have done. So what we did, we have done one study in collaboration with AIIMS Raipur. Okay, where we have collected our own data, like real time, our own data was collected. So the study was approved by Institutional Ethical Committee of NIT Raipur and AIIMS Raipur. and we collected a multimodal data set. Now, what happened if you see all the public databases which are available online, they contain either mammogram or they contain either ultrasound. Okay, so most of the studies, if you see maybe 5-6 years back, most of the studies were most of the CAD systems, computerized diagnostic systems, they use either mammo or ultrasound, not both. But it is reported in literature that if they are used in combination, the performance will be slightly better. The performance of radiologist will be slightly better. So similarly, we assume that performance of CAD systems will also be better. Okay, so that's why we collected a multimodal data set. So collecting such real-time data sets is very difficult because we cannot ask the patient unnecessarily to go for mammogram also and ultrasound also. Okay, it can be done only if required. So that's why we could get only 31 patients which underwent both mammography and ultrasound. And I think it was 31 or 32. I do not exactly remember. I think it was 31 or 32. So out of these, 25 were found to be benign and 18 were found to be malignant. Now, also of these patients, there were multiple images were taken, not only one images. So ultimately, our database included of 86 images, 43 mammogram and 43 ultrasound. Okay, multiple views were taken. So we did some unimodal studies also, and I'll just discuss this one study in which we did this. We did only using ultrasound images. Okay, so we took the ultrasound images. We did some pre-processing. So if you see this, it is written as speckle removal, noise removal, and ROI extraction. So we remove the speckle noise and we take, like if this is the image and this is the tumor, we extract only this area. So this is called as region of interest. Then we did feature extraction and normalization. Lot of features were extracted. I think I have not included those things in here because of the limited time. But this is the paper which we published, so that can be seen. So we extracted lot of features. We did feature normalization. We selected the features. Then data was divided into two parts: training data and testing data. Okay. This training data was first given to fuzzy C-means clustering. Okay. Now, if you all might be knowing, this fuzzy C-means clustering is an unsupervised method. Generally, we don't use unsupervised methods in ML and DL models for classification. Okay, because unsupervised methods are used when the label is not available. But in our case, label was available. We knew which cases are malignant and which cases are benign. Okay, so FCM is why we used unsupervised machine learning. Basically, we have a training data, and this training data was first given to fuzzy C-means clustering. Fuzzy C-means clustering will just give two groups because the number of clusters were set to 2. So this is group 1 and this is group 2. Then we analyze these two groups. The group which had maximum number of benign samples were labeled as benign, and the group which had maximum number of malignant samples were labeled as malignant. Now there were some benign malignant, because it is a clustering approach. There were some malignant cases here and there were some benign cases here. So we eliminated those samples, considering them as faulty samples. Maybe the process was not accurate or some issues were there with those samples. So we eliminated those samples. So the purpose of using a unsupervised learning in this particular pipeline was to identify faulty samples and eliminate them, so that the overall performance of CAD systems can be improved. So if you see here, what we have written here is, so we get two clusters, then we match the sample ground truth with the cluster category. Okay, so this is cluster 0, this is cluster 1. If cluster 0 means benign, 1 means malignant. So if this cluster category matches with its actual category which we have, the label which we have, then that is kept. If it does not matches with the actual cluster, we eliminate it. So I have written here, eliminate the doubtful samples, mismatched cases. Here also we eliminate the doubtful samples. And after that, we get a refined training data. This refined training data was then given to backpropagation neural network for training. So we get a trained neural network model, And then the test data was given to this trained neural network model, which was for categorizing the test data into benign and malignant. Okay. So the purpose of using combination of supervised and unsupervised. Backpropagation is a supervised technique. FCM is an unsupervised technique. The unsupervised technique was simply used to eliminate the doubtful samples so that we get a good trained model, the model which is trained with only good samples. And then we apply the test data and we evaluate whether it is correctly able to classify benign and malignant cases or not. And we were able to report accuracy of like 94%, high accuracy of 94%. Okay. Then we also did another unimodal study. This I will skip. I'll go to directly come to the multimodal case. So as I told you that in this AI models, combination of ultrasound and mammogram may give a better result. Now I also want to tell you that this study was performed on online data, not the data which we collected. Okay, so I cannot compare the result of this model with my multimodal approach. This was done on online data. Now we come to the multimodal approach, in which we did individually using ultrasound, individually using mammogram, and also combination of both. Okay, the process is same. We did pre-processing to remove noise and artifact. Then we do data augmentation to increase the number of images because the data size was small. It had very few images. So we have to do data augmentation to increase the data size. Then we go for feature extraction. We go for feature selection. And we also did some statistical analysis like Mann-Whitney U test, students T test to select only the significant features. Okay. Ultimately, like you can say that this is also like feature selection. Okay. Because if I apply say Mann-Whitney U test and only those features which are significant, are taken further. Others are removed. So we have 42 features. We further took only 36 features because the remaining features were not statistically significant, which means that they were not able to categorize benign with malignant significantly. Okay. Then we did classification only using ultrasound. Same process was used for mammogram also, but here out of 42, we took only 23 features because only 23 were significant by using Mann-Whitney U test. And then we did classification using mammogram. And then we did multimodal. In multimodal, we combined the features extracted from ultrasound with the features extracted from mammogram. We combined them and then we did the combined classification. Okay. So same steps were used. Okay. We did normalization, for feature selection, we did statistical significance analysis. We used Mann-Whitney U test. Okay. And these are the features which were extracted. Okay. So first order statistics like mean, median, skewness, kurtosis. Then we have this energy contrast. So a lot of features are there in literature. We can extract those features. And these are the classifiers that were used. Support vector machine, KNN, discriminant analysis, logistic regression. And this was the result. So by using, so this is a multimodal system. Okay, so this was by multimodal system, the highest accuracy which we were able to get was 98%. And by only mammogram alone, we've got 93% highest accuracy. And by only ultrasound, we got 91. So 91 was the highest unimodal ultrasound. 93 was the highest unimodal mammogram. But when we use both ultrasound and mammogram, we get accuracy that is equal to 98.84%. So we were able to prove what is reported in literature is that by combining ultrasound and mammogram, the CAD systems can detect breast cancer with higher accuracy. Okay, and that was a machine learning approach. We also did a deep learning approach. In deep learning approach, this is again a multimodal image, mammogram as well as ultrasound. This is a typical CNN architecture. Okay, this is a typical CNN architecture. It has layers like convolutional layers. I'll not discuss those. Okay. And finally, there is a layer called as fully connected layer. So this was a hybrid approach kind of thing. Why? Because what we did is, in CNN, generally the last layer is softmax layer, which is used for classification. But we eliminated that layer, and the features that were extracted by CNN at fully connected layer were given to LSTM. LSTM is another classifier. So this technique was combination of CNN plus LSTM. CNN was used for feature extraction and feature selection, and LSTM was used for classification. This type of architecture. Okay. So LSTM was used for classification into benign and malignant. So here, if you see, we did feature extraction. Mammography gave us 100 features. Ultrasound also gave us 100 features. When we combined the features, we got 200 features. When we did statistical significance analysis, that were reduced to 165. So 200 got reduced to 165. Then it was given to LSTM. And finally, we did classification using LSTM. Here also, we were able to get more accuracy using multimodal approach. But the difference was not as good as in machine learning, because unimodal also gave good performance, 97, 98, and 98, 99. So maybe because the data size was quite low, if we evaluate this on a very large data set, a more significant difference could be observed. Okay. So I think with this, I will end the presentation. If I'll take some questions. So this work was primarily done by me and one of my scholars. So I would like to acknowledge my supervisors, Dr. A.S. Toke and late Dr. Keshri Verma, because I did this work under their guidance during my PhD. And after that, the most of the multimodal work was done by Kushangi Atre. She is my PhD scholar. Already she is passed out. And this was also done in collaboration with AIMS Raipur. So some of the work was also funded. So I would like to acknowledge the funding agencies also. I'm now open to questions, if any, from the audience. I was
Thank you
little
very
fast
much.
because one hour is little difficult. I like to accommodate everything in 1 hour is little difficult. So I was little fast. So if any questions are there, please let me.
Yes, thank you very much, Professor Bikesh Kumar Singh, for such an enlightening and thought-provoking lecture. So your presentation really provided a valuable insights into the artificial intelligence biomedical image processing. So we shall now move on to the question and answer session. Participants, those who want to ask any question, you can raise your hands, or you can post the questions in the chat box. Sir, it was really a very wonderful session, because you have made everyone understand each and every step very clearly. In fact, I have no knowledge of this before, but now at least I have an overview. I know how this image processing is done. So it was a very nice session, sir.
I think one participant, Bhavna, you want to ask something? Thank you, madam. Thank you.
Yeah.
Please go ahead. Okay, one question is there. After mammography, is feeding to the baby harmful? Actually, so the clinician will be the right person to answer this question. But it's obvious that uh, uh, generally in pregnancy stage, these tests are not done. It may be harmful. But after that, it may not be harmful for the to the baby, but clinician would be the best to answer this. Okay. Bhavna, can you
Hello,
type
good
the
morning,
question
sir.
if you are? Oh, okay. Good morning. Yes.
Yeah, good morning, sir. I just want to ask, you have chosen uh, mammography and ultrasonic, both the model, I think. If
Yes.
we can
Yes.
combine all three as to correct the accuracy, MRI can be also be included into this?
It can be included, madam. Like we are working on breast elastography currently. The problem is that getting the data would be very difficult because like MRI is a very costly technique. Okay, compared to mammogram and ultrasound, MRI is a very costly imaging approach. So, asking the patients to go for 3 imaging modalities would not be practical actually, from cost point of view also, and uh, MRI also takes a bit more time compared to ultrasound and mammogram. It is time. MRI is time taking also. Like, but and CT is not time taking, but it has lot of radiation. So generally,
Yes, sir.
I would say it is not practical to include MRI, but definitely like MRI can be used like in cases where like there are certain cases where MRI is done uh, because it is required. So if we get that kind of
Yes.
data, definitely it may improve the accuracy. Like we are going for breast elastography. Elastography would be better because it is also ultrasound based. It is not harmful, and sometimes it is done in a routine procedure. Mammogram and ultrasound are routine procedures. That's why we included it.
Good, sir. Thank you so much, sir.
Well,
Thank you, sir. So, as a token of our sincere appreciation and gratitude, we are delighted to present a digital memento and certificate of appreciation to our esteemed speaker. Uh, uh, may I ask Dr. Gaurav Kumar, HOD, sir? Yes.
Thank you, sir. Thank you for accepting our invitation. So, on the behalf of organizing committee and our department and school, as well as university, I am mm, grateful to present uh, this e-memento to you. So, kindly accept it, sir.
thank you.
Thank you, sir.
Yeah. Thank you, and thank you for the opportunity. Thank you for the invitation. Thank you. So I think I can leave the
Yes,
meeting now.
yes, sir. Yes, sir.
Thank you.
Thank you so much, sir.
So, Vivek sir has joined.
Yes.
Okay, sir. It is now my pleasure to introduce our next distinguished speaker, Dr. Vivek Kumar, Assistant Professor in the Department of Biotechnology at SRM Institute of Science and Technology. Dr. Kumar obtained his PhD in chemical biology from the Indian Institute of Technology, Delhi, and subsequently pursued his postdoctoral research at Carnegie Mellon University, USA. His interdisciplinary research focuses on understanding biological barriers and developing innovative molecular delivery strategies using peptides, nanomaterials, and fluorescent-based analytical tools. His work encompasses cell-penetrating peptides, antimicrobial peptides, nano-enabled delivery systems, fluorescence spectroscopy, biointerface engineering, and plant biotechnology, with applications extending across healthcare, agriculture, and sustainable technologies. Dr. Kumar has published in several prestigious journals, including Nature Nanotechnology, Angewandte Chemie, Applied Microbiology, by Microbial Journals. We are delighted to welcome Dr. Vivek Kumar to our FDP. We now request you to kindly deliver your lecture on understanding biological barriers through peptides, nanomaterials, and fluorescence tool. So the virtual platform is yours, sir.
Thank you. Thank you, everyone, for inviting me for this International Faculty Development Program. Today, I will be discussing something about the understanding of the biological barriers through the peptides, nanomaterial, and fluorescent tools. These are the three components that I have been trained on, and I'm working still on this. So, the one question that comes to our mind is, why do so many promising drugs like the nanoparticles, proteins, and the genes fail to reach their target? It's because every one of them must have first to negotiate with the biological barriers. Is it? So that's why I've designed the topic in such a way that will encompass every components of this one. And that's the understanding the biological barrier through the peptides, nanomaterials, and the fluorescent tools. Why do biological barriers make therapeutic delivery so challenging? The reason is the biological barriers we have, the carrier molecules are diverse in nature. The proteins, nanocarriers, the DNA, the genes, everyone are of different nature. Along with that, the first challenge is never the target. It is reaching the target. Once you reach at the target, then only it will cross. And once it reaches the target, the barriers are of diverse in nature. The cell membranes of the mammals, the plant protoplasts, bacterial membranes, the organelle membranes, and the blood-brain, and many more. All the barriers are of different kinds, posing different kind of challenges. So one biological barrier and many challenges. What are they? Although the cargo changes, the fundamental challenge remains the same. Every delivery technology ultimately becomes a biological interface problem. This is not just a drug delivery problem. It is the problem of every field, like protein delivery or precision, CRISPR-Cas genome editing, gene delivery, nanomedicines, agrochemical delivery in agriculture, everywhere. So regardless of the cargo, successful delivery depends on understanding the overcoming biological barriers. Based on this, we will understand the biological barrier by three ways. First, by peptides, we will understand how they are crossing. Second, the fluorescent probes. It will help us to understand the biological barrier. And the next one, we will engineer the carrier molecules, like a nanocarriers. That will solve the biological delivery solutions. The agricultural problems will be solved, the therapeutic problems, diagnostic and nanomedicine as well. So let's first see about one of the example of peptides. The one peptide I'm showing you is a peptide called tachyplasine. It has been isolated from the horseshoe crab, marine organisms. Marine organisms are quite well known for having proteins and peptides which are sustaining into the harsh environment. So these are 17 amino acid longs, and the two anti-parallel beta parallel sheets are there. That's serum. The 4 cysteine amino acids provides a disulfide bond, providing them a serum stability. So then comes the cell membrane. Anything which is acting on a membrane or membrane active. Here we are discussing the peptides, so membrane active peptides. If the membrane active peptides acts on a controlled interaction in a very precise way for a short duration of time, like 1 hour, they are capable of delivering the DNA, any gene, or a proteins. Like here, the tachyplasine, it delivered the EGFP protein, a DNA, which entered inside to a live Leishmania donovani organisms. It again showed how it can deliver the GUS protein into a mammalian HeLa cells. And when the interaction time is more, we have a more duration of time to act on the membrane. It creates a pore-like structure, leading to the release of all the, let me just have a pointer that will easy to point. That will help us to create a pore-like structure into the organisms, while not damaging any of the host cell membranes. It is because they are made up of different components. The parasitic membranes, the biological membranes of the host cell, all are of the different kinds. These peptides have a specificity towards the host cell, which are made up of phosphatidylchlorine or sphingomyelin, whereas the parasites, its cell membrane, it is a mastigot, which is inside a macrophagic cell, it will be of a glycoinositophospholipid. And if it is the promastigotes, it will be the polysaccharides, lipophosphogycan. So that's why the interaction time is the more important, how much time it is attaching with the membrane. We also know that most of the peptides suffer from the stability-related issues, toxicity-related issues, and the many more issues like immunogenicity and other things. Most of the time, if they are suffering from these kind of issues, should we throw them? I think no. So what we have to do is, we have a natural peptides. What we will do, we will engineer the peptides. We will modify the peptides and we will form a new group of the peptides called peptides. What they do, how they are different, the peptides are a variant of peptide. Where in general, the peptide have a functional group R, which are attached with the alpha carbon, while a variant of a peptide where these functional group have been attached with the nitrogen into the peptide bonds, giving a more stability towards the serum and a longer lifetime into the serum, making the peptides availability for longer time. So longer interactions, more antimicrobial activities. What it does, it improves stability, it tunes the hydrophobicity, charges can be tuned, selectivity can be tuned, and toxicity can be tuned. So what we did, we have just modified the peptides into 8 variants. What are these? The lysine. The lysine are N-substituted lysine now, because these are the peptides, not the carbon-substituted functional groups. Then comes N-substituted phenylethylene glycine. It's another variant of the amino acids that provides more stability into the serum. And this chain was created, polymer, dimer, and trimers. At the same time, we are also known that the halogen groups, chlorine, fluorine, bromine, bromines, all are known to be very toxic towards the pathogen. They have antimicrobial activity itself. So what we did, we tagged the bromine amino acid, bromine with these amino acids to prove the antimicrobial activity and enhance the antimicrobial activity. We tested them onto the Leishmania donovani or Bob cell sign, and we found that in just 4 hours of the treatment time, the probability of the Leishmania promastigote cells drops by 50% at a concentration of just a mere 1 and 3 micromolar. Whereas when the bromine amino acids was tagged with them, we found that it have a more antimicrobial activities. It showed that when a bromine tagged modified peptides have activity starting from 1 micromolar, it's a very small concentration, and its activity is optimum at 3 micromolar, whereas the host cells have almost negligible toxicity at the desired concentration we are treating them. So what this says that if one naturally occurring peptides can interact with the membrane in such a different way, can we engineer the peptide specifically for membrane transport specific to the organelles? Can we do that? Yes, it can be. Engineering membrane interactions solves only the first problem. Once inside a cell, another challenge will emerge. How do we guide the cargo to the specific organelles? And that's why the another work which says that how to modify the peptide for organelle targeting. And that's that we have a natural chloroplast targeting sequences or signal sequences, transit peptides, or many more names they have. These are like a postcode. That postcode directs the protein, the cellular translation proteins to the specific organelles. So what we did, we isolated the sequence of all those transit peptides. We screened them on the basis of cell penetrating properties like more positive charged, more hydrophobicity, less PI, and the other properties of the cell penetrating peptides. And that made the peptides cell penetrating as well as the chloroplast targeting. So what we had, we landed with the 2600 approx transit peptide sequences. Screening them on the basis of cell penetrating reduced the number to 20. And on the basis of membrane interaction studies, the number was reduced to 8. Out of them, 1 was tested. And what we found, the peptide enters the membrane in a concentration-dependent manner. You increase the concentration, more peptide will enter. But at the same time, the question will come. If we keep increasing the concentration, the toxicity will happen. So we must check toxicity. So at that 30 micromolar, we have a sufficient number of peptides inside, whereas no significant toxicity on the protoplast cells. These were visible through the epifluorescence microscope. Then we had a question. What is the minimum concentration and the minimum time required to have this peptide to enter inside? So we tested it and we found that the peptide start internalizing from 1 micromolar concentration in just 5 minutes of the time. At 3 micromolar concentration, sufficient number of the peptide have internalized in just 5 minutes of the treatment time. Why the 5 minutes of the treatment time is more important? Because in agricultural system, when we spray the agricultural chemicals, the major challenges are, first, they will not stay on the leaf because of the waxy layer. They will fall down. So they will have a very short duration to act. If the peptide acts within the 5 minutes, we can make sure by allowing some kind of a surfactant to let it stand for 5 minutes at least and let this peptide work for its activity. So that's why this 5 minutes is more important. Then what we did, we tagged this peptide. Ultimately, a peptide needs to deliver something. We are not interested, we are not working on the peptide as a blocking agent. We are using this as a delivery agent, delivery vehicle. So first, we tagged the peptide with a protein called DNase1 and tagged the fluorophore with that DNase protein. We did the same experiment onto the protoplast and we found that protoplast enters inside the cell, do not goes into the nucleus. You see this space is blank. There's nothing inside. So do not diverts, directly delivers the proteins. How we can say that protein is delivered? Because the fluorophore was tagged with the protein, not with the peptides. Confirming this, but the second approach for validation is required. So what we did, we performed the qPCR. Why we performed the qPCR? Because I told you the DNase1 was tagged with this peptide. So what the DNase will go, it will go inside to the chloroplast and cleave all the chloroplastic DNA in a non-specific manner, leading to the reduction of all the genes, destruction of all the chloroplastic genes. So we choose rubiscosines. What the rubiscosine will do, it will be responsible for the photosynthesis. And we found that when we deliver that, the fold change increases. And that change in the fold change proves that the peptide have delivered the DNase. That DNase have deleted all the genes, degraded all the genes. Again, we cannot see the protoplast everywhere. Protoplasts are the ideal things. It is the plant which have a number of barriers in it. So we tested it onto the isolated wheat leaf. And we found that though the fold change is less, but still it's reducing the DNA activity, the gene activity of the rubisco. Test proving that a peptide can deliver the protein. We can modify the peptides according to the membrane environment. How this peptide is more important because this condition, it crosses the 3 biological barriers. First, the cell membranes. Second, chloroplastic membranes, and third, thylakoid membranes. If you see the pictures here in this playing video, you will see that the thylakoids can also be possibly visible using this confocal microscopy. So moving forward, we have now that we have a protoplast, but as we know, protoplasts are not the ideal things. We have to work on the whole intact plants. And what are these? The intact plants have 3 important barriers. The waxy layer over it that make the water molecule slip out. Otherwise, all the chlorophyll, water will go inside and solubilize all the chlorophyll, and we will see after rain, all the plants are white in color. That's the nature have prevented them using providing a waxy layer. Cuticle. a very hydrophobic layer present below the waxy layer, and then comes a natural cell wall. Combining all, the maximum size exclusion limit of these three combining is 5 to 20 nanometers. So anything which is beyond the size of 20 nanometer cannot cross the leaf surface, cannot cross all the three barriers. Sometimes some scientists have shown that it can be increased up to 50 with a certain conditions. So what's the advantage? We cannot work on anything. Everything work is on a simplified system, but the whole plant is more complex. If we want nanoparticle to cross a biological barrier, simply synthesizing is nanoparticle is not good. So the first interface they encounter is biological barriers. So we have to modify the nanoparticle that will cross all the three as well as the cell membrane biological barriers. So that's why the whole plant barriers. So what we did, we choose a nanoparticle, we synthesized it, that will have a more surface interacting capability with the leaf. Then we choose a R9 peptide, a polyarginine peptide, or you can say that nona-arginine peptides. We combine them because this will surface modify it. This will functionalize the nanoparticle surface. And then we tested it onto the wheat plants. And what we found that when we treat them onto a protoplast, it internalization increases about 2.5 times. The same was also tested using the fluorimetry. There was a small tracker molecule vanadium which was incorporated into this nanoparticles for a tracking purpose that will help to do the ICP-MS waste identification of the nanoparticle or tracking wherever they are localizing, they are moving, everything. In the second approach, the nanoparticle, they had instead of the tracker molecule, the fluorophore that helped us into the confocal microscopy-based experiments and the other fluorimetry-based experiments. And what we found that the nanoparticle itself attaches after several rounds of washing of the treated leaf. 70% of the nanoparticle remain adhered onto it. But when the nanoparticle was formalized or functionalized with the R9 peptide, the association increased by 15 times more. And it's not only the association, it is the translocation was also increased from 6% on a non-functionalized nanoparticle to 16% on a functionalized nanoparticle. I just reminding here that the functionalization here, the peptide were not tagged with the nanoparticle. They were co-applied, like A plus B. And it was also interesting to note that it reached to the root surface as well. So you are applying on a foliar, it is reaching to the root. Why we are more interested in the root? We know that there are many infections on the foliar. Why should not consider this one? But there are two conditions which are very important. I will point here example of the citrus, a major example where the major food crop in the US in the Florida region is citrus, and they are suffering with a disease of a bacteria. It enters inside the plant, citrus plant, and block the xylem of this whole plant in the crown region, leading the plant to die in 2 to 3 years. And the second example is, the well-known example is the nematodes. They are residing in the soils. If you apply the nematicides into the soil, you will have to apply a ton of the chemicals. That's have ecological disadvantages. What if you supply the nematicide using the foliar leaf, when the nematodes will approach the root, it will come in contact and get killed. So that's why the root internalization is very important. And we found that our internalization is also on the stem. So how it is beneficial for the stem borers in the rice, in the maize? They are helpful. If the insect will approach to the stems, the nanoparticle with the drug-loaded insecticides, they will kill them. So peptide functionalization increases the nanoparticle association with the plant cells and enhances the transport beyond the treated leaf. Now comes how it enhances. What are the mechanistic pathways? The mechanistic pathways, first of all, how the functionalization appeared onto this. So first, when we applied the nona-arginine peptide with this, the charge reversal happened. You see, the nanoparticle was almost 48 minus charge. But when we applied a peptide, it charged totally converted into the positive side. And also, it is aggregating. So although the peptide coating increases the hydrophobic diameter, the reversal of the charge surface, and the membrane active properties of this nona-arginine peptide dominate the interaction with the whole plant cell, leading to the enhanced uptake. So we have to prove this. So how it was proved? It was proved using a confocal microscopy. This time we used a fluorescent probe, and this fluorescent probe, we saw that when we just treat the protoplast with a nanoparticle, only one few or two nanoparticles you will see here. But when the nona-arginine at a 10 micromolar concentration was co-applied, the nanoparticle internalization increased. The increasing concentration leads to more number of nanoparticles inside the protoplast. What about the whole plant? We do the cross-sectioning of this. This time we used a vanadium-containing nanoparticle and from the X-ray fluorescence. How this is different? So this is an orthogonal approach. We are approaching the same target from the different sides. So we treated the cells with these vanadium-containing nanoparticles. We used the formulation with the R9. Then after we do the microtomy and perform the X-ray fluorescence. Here the phosphorus represents the cell membrane. Sulfur represents the protein contents of that area, the section. And the blue color represents the vanadium-containing fluorescence. It was found that when nanoparticle was treated, few amounts of the nanoparticle enters inside. But when the nona-arginine was used as a formulation, more number of nanoparticle internalizes into the protoplast, suggesting that the nona-arginine, why we are more concerned about the nona-arginine? No, peptides are very costly. So here we propose that if we will use some positive charge containing polymers are very easy to synthesize and very cheap in cost. So if we use some polymer containing positive charged and use this as a formulation strategy, that will enhance the foliar internalization and the translocation. So that will be a good asset. So what the peptide delivery determines? Unlike X-ray fluorescence, if you see this X-ray fluorescence, it directly detects the elemental signature. It's a better way of confirmations, eliminating the ambiguity arising out of the fluorescent related studies. These results also shows that peptide engineering can modify the nanobio interface and provide a strategy that overcome the biological barrier interactions. This experiment will also show you that how a nanoparticle size increment in this one. We have seen that when the nona-arginine was provided, the size increment was several fold, almost 4 times. Even the size increments is not only the single determining factor, it is the surface chemistry, how the peptides are coating the nanoparticles. That is also determining the factor. So all these combinedly surface engineering, not the particle synthesis alone, determines how the nanoparticle interacts with the biological membrane. By functionalizing the nanoparticles and the membrane active peptides, we have changed the biological identity and enhanced the translocation and association as well. Then comes the co-application. Till this, we have applied that. What if we will functionalize the nanoparticle with the same peptides, perform the same experiment again? So what we did, we first assumed that peptide conjugation will always be superior. Before this experiment, it's a general idea. Add a peptide to the surface, it will bring that nanoparticle or some carrier molecule, whatever the requirement or wherever the requirement it have to deliver. But this nano formulation with co-application showed that co-application is better compared to the nano formulation strategy. So what we did, we designed a LDS nanoparticle. And this LDS nanoparticle was loaded with a fluorophore called Fluo4. And we did functionalization as well as the co-application. And we found that when we did the functionalization, there was no fluorescence detected in this panel. But when co-application was done, the nanoparticle get associated with the protoplast membranes. This surprisingly simple co-application produces substantial greater intercellular delivery. We performed a single cell analysis and performed the quantification as well. because in this panel it was really difficult. So what we found, the co-application leads more nanoparticle to enter inside the cell, and it was several fold higher than the functionalized or non-functionalized nanoparticles. So what they said, this suggests that peptide dynamics rather than the permanent immobilization may be the critical for crossing the biological barriers. The mode of peptide presentation, not just peptide identity. So it's very important. It is the mode of the peptide presentation is important, not the peptide identity. We have used the same R9 peptide, but we saw the different result of the intercellular delivery. This suggests that the peptide dynamics rather than the permanent immobilization may be critical for membrane barrier crossing. So looking beyond the peptide internalization, we have seen the peptides internalizing, they are entering inside, they are exploring everything, but they never say anything related with the membrane. We have a peptide, it will enter inside and it will deliver something, but no information about how they interacted with the membrane. Conventionally, where are the peptides? We can say, did the peptide enter? Certainly we can say by fluorimetry or confocal microscopy. Where the cargo is localized? Yes, we can say. Uptake efficiency we can determine, and the immobilization pathway we can determine. But how the membrane responded during this transition time when the peptide was interacting with the membrane? Did the polarity of the membrane change? Because why polarity is important? The nanoparticle we have designed that will change its surface properties. If the polarity is something different, we will change the nanoparticle chemical composition. What was the hydration effect? We know that the nanoparticle have a corona outside, a hydration layer. If that hydration will change with the peptides membrane surface, we will have two different chemical compositions to change the corona of the nanoparticles. How the lipid packing changed, how the orientation, it will be deciding the orientation, and what was the mechanism of interactions. None of these was told using the conventional peptide labeling. So that's why peptide labeling tracks the peptide. Membrane probes report the membrane responses. So what we did, we delivered tells us whether the peptide transport occurred. But a fluorescent membrane probe will explain why it occurred. So what we did, instead of labeling, we labeled the whole cell with a fluorophore. And a well-known fluorophore called ANS, 1-alanino-8-naphthalene acetic acid, is a well-known probes usually used to determine the hydrophobic change into the protein-related studies. It's an environmental sensitive probes. What it does, it goes and binds with the hydrophobic patches. The word is very important, the patches. It do not bind with the hydrophobic amino acids. It binds with the hydrophobic patches where more than hydrophobic amino acids are present. So based on this hypothesis, what we have an assumption that this fluorophore will go and bind with the lipid raft, a very common used words into the cell membrane surface. Lipid raft, and when we will apply the nanoparticle or a peptide or any biological carriers, the lipid raft will change, the lipid packing will change, the hydration property will change, and that change in the lipid raft property, the ANS will more bind or lose. That losing up and down of the ANS signature will give rise to some signal about the membrane, whether they are interacting with or not. The second is that if you tag a fluorophore with the membrane peptides for tracking, it will change its biological and physiochemical properties. So that's another limitation of tagging this one. So we isolated protoplast, we treated the whole protoplast with the ANS. And what we found that when a tagged peptide was applied onto a ANS treated cells, there was a peak shift. Peak shifted to the left side and upward, the more quantum yield. Whereas in the M-tagged, it is a mutated form of a tag where the positive charge has been reduced to make the peptide less interacting with the membrane. It remained same with the protoplast fluorescence. Whereas the other parameters, control parameters, do not matches with the positive controls, saying that indeed the fluorescent molecules can be utilized for tracking the cellular membrane interactions. What about the animal cells? Most of the people's 90% of researchers are working on animal cells. We indeed also worked on animal cells, and we also found that With the animal cells, the R9 peptide, the polyarginine peptides, they also interacted in a similar way. And there is a quantum change. Quantum yield has been increased. The peak shift increase, peak shift left or right. Quantum up and down fluorescence peak shift will say you the membrane interaction capability with this membrane. So tracking the membrane is not the same as understanding the membrane. What it says, the different peptide, the TAT, R9 and the TAT, the different cell-penetrating peptides produce a distinct membrane responses upon the peptide treatment. And the membrane perturbations, the change into the membrane lipid structure or compositions, is the fingerprint distinguishes the peptide behavior. So this behavioral can be utilized to distinguish a large scanning, a high throughput scanning of the cell penetrating peptide. Suppose there is a peptide lab who is working on a kind of a hundreds of peptides. If they will keep going with tagging all the 100 peptides with the fluorophore, the cost will go higher, the experimental time will go higher. By this method, what they can do, synthesize a peptide and just go ahead. This whole experiment is just of a 15-minute duration. You have to take a cell, treat it with the fluorophore ANS, add a peptide, the designed peptide you had, wait for 15 minutes, go with the fluorescence plate reader, and will give you the 96 data in a single 5-minute experiments scanning. So you have scanned actually the 96 peptides in one single scan. So until now, we had only answered one question. Did the peptide enter the cell? ANS still yes, it enters. It's a different questions. Then comes what happened during the entry. Though because of the time lag, I thought that 1 hour will not be sufficient. I thought to remove the remaining part. So we have used dual dye and triple dye systems. So in general, the ANS binds with the hydrophobic patches only. What about the inner leaflet of the membrane? If a peptide is coming in contact with the membrane, starting the internalization and getting stuck into the membrane, that will also give the signature. So how you will differentiate or distinguish between that one? For that, we have used the second dye system and the third dye system. That will go and bind with the membrane and interact with them, will give the different signature, the time-lapse fluorescence spectrometry, the PC analysis, all will give you the data. I am not including those papers because it was just accepted last night. So every peptide produces a distinct membrane perturbation and a fingerprint. And interestingly, the peptide with similar uptake can induce a very different membrane response. We have seen that the R9 and the TAT both are the cell penetrating, but they have a different peak shift on the TAT, but no peak shift onto the R9. Only peak increase onto the R9 was observed. And this means that the uptake alone is not enough to understand the mechanism. We have to work more on this. ANS showed that membrane itself can report the peptide interaction, but as a whole only. Could we separately observe? Yes, we can observe. We're doing a dual dye system, triple dye systems. So what are the future engineering? How we can engineer the barriers so that the more therapeutic molecules will go inside. We have a better understanding of the membranes, and the more smart barrier molecules, peptides will go enter inside that one. So what it says that the next frontier is not simply crossing the barrier, but designing them with precision delivery. It is not the carrier. We need to understand it. And that's why I will say you the take-home message for you, everyone, is that biological barriers are not an obstacle. They are an intelligent interface that can be studied using the peptides, nanomaterials for engineering them, and fluorescent probes for understanding them. So we will understand it today and we will enable it tomorrow. Thank you for your attention. Though this work was done by me, but it was a collaborative work done on a different institutions. My IIT Delhi, Carnegie Mellon University. I joined the Carnegie Mellon on a BSA project, and the other collaborators were the Astrid from the France, Hagai from the Volcanic Center from Israel, and Zivoni from the ESRF Grenoble. So I think I'm over from here. I will open for a few questions if anyone have. Thank you.
Thank you very much, Dr. Vivek Kumar, for delivering such a fascinating and insightful lecture. So, your presentation beautifully illustrated how peptides, nanomaterials, and fluorescence-based technologies are revolutionizing. So, now I would invite questions from the participants. If you have any question, you can kindly raise your hands or submit your question in the chat box. Hello, sir. I would like to ask one question. Can you suggest something regarding how can the you mentioned, right, the stimuli responsive behavior of this fluorescent probes that are we are attaching with the peptides. So can you say something on this, like how will the stimuli responsive behavior will happen?
In the nanoparticle engineering, what we do that we use some kind of a lipid molecules, like those composition of the lipid molecules that will be stimuli responsive, stimuli in sense of the pH change. If you apply onto the leaf surface, they have one pH. Once it enters inside the cell, the pH changes and the cargo will break. So that's the one of the way we use the stimuli responsive. Second stimuli responsive is once it will bind with some kind of another carrier molecule, like there are hundreds and thousands of the protein molecules inside a cell. So you tag a carrier molecule that will go and bind with that. The time it will bind, because of the freight kind of interaction, it will break with the carrier. Because if you have a carrier with a target molecule, if both go and bind with the target molecules, it will not work accordingly the expectations you will have. You need the carrier to be left out.
Right.
So those kind of interactions we are using.
Okay. So thank you very much, sir, for your talk today. And as a token of appreciation, we are pleased to present a digital memento and certificate of appreciation. May I request Dr. Gaurav Kumar to kindly present the digital memento, sir.
Okay, thank you so much, ma'am, Rupta ma'am. As I'm fortunate to present you, sir, a digital memento on behalf of our Department of Biotechnology and Bioengineering, School of Bioscience and Technology, Galgotia University. And we are grateful that you have accepted our invitation to deliver such an insightful and informative talk. So kindly accept this digital memento from our organizing committee.
Thank you for this. I like this innovative idea.
Yes.
Thank you for this one.
Thank
Thank you for the committee, every members.
Yes. Yes, dear participant, if you have any query, you can unmute yourself and you can ask if you have any question.
I think there is one question. What are the biggest challenges in translating bioengineering research into commercial biopharmaceutical product? So it depends on what kind of biopharmaceutical product you are looking for, like peptide or nanocarrier-based engineering. For peptides, I told you the major problem is the serum stability. Though we have some approaches like we are just adding some cysteine amino acids to make it more, we tag some amide groups to the end so that the cleavage will be less. But peptides are always problematic. That's why we shifted from peptide to peptide research, a kind of a variant of peptide, a mimetic structure. Biomimetic, we can say that. So in that conditions, we use some synthetic amino acids. They are more resistant towards serum proteases. They are less immunogenic as well. And the sizes are controllable, controlled according to our expectation. In peptides, in biotherapeutics, mostly we think that we will go for 11 and 12, but in general it increases to 18, 20, and 25. The longer the peptide, the more immunogenic properties it will have. So that's why this peptide gives us an opportunity to make it 3 or 4 batches and just make it twice, thrice polymer kind of things. It will just less immunogenic in nature. So we have proved this. So our research group in a collaboration in Stanford, she is Annalise Baron. She have a great experience with these peptides, and she is commercializing these things onto the eye disease, eye and the lungs. If someone is interested, they can look into that. Dr. Annalise Baron from Stanford.
Sir, I have one question. This is Dr. Charu Gupta. Sir, if I have certain peptide and I want to cross-link, get it cross-linked with some matrix, so are there any commercial groups available which provide that service, or we have to do it in lab? Actually, I'm new in this field, that's why I'm asking.
See, now you are asking the kind of industry kind of questions. I'm not industry expert, but I will say you one thing.
No, sir, I understand, but this is the kind of thing that sometimes students also ask, and I'm new in this, so that's why I'm asking.
No, it's okay. It's okay. So there are two aspects here. First, the cost aspect. If you go with the industry persons to let it tag them for you, the cost will go beyond our expectation. It will go to the 1 lakh for one tagging peptides. It will give you only 4 mg or 5 mg. So I will recommend to just work on self on the chemistry side. Like in general, we use the EDX based click chemistry to just do the tagging and other things. It highly depends on what kind of matrix you are using.
Okay.
And it is not the tagging only. The second question is,
suppose if I'll have to study more.
Yeah. Now I am just pointing one more thing. If you will think that you are tagging a peptide, it's a very broad word. Tagging, how the alignment will matter. Head to head.
No, I'm not. I'm not talking about tagging. It's like I want to cross-link it to the matrix so that it can be used for certain applications. So I'm not trying to tag.
That's what I'm
just.
That's what I'm saying. Actually, the cross-linking is a very difficult process. If you will cross-link now, and suppose the peptide is cross-linking from the mid, it will lose its bioactivity.
Yes.
So you will have to take.
Yes, sir.
And I think we have someone.
I understand.
Okay. We have one more question. Thanks. Welcome. So we have one more question. Do this peptide alter any channel in the membrane? They can. You can design the peptide that will bind with the channel membranes, membrane channels, or any transporters. I think I have just answered that one. No?
Yes, sir. Yes, sir. Okay, thank you. Thank you so much, sir.
Thank you. Thanks for this invitation.
Thank you. Thank you so much.
Should I leave this?
Yes, sir. You can.
Thanks.
Thank you, sir. Sir,
yes.
Dr. Najwa Mohamad, sir, is here.
Yeah, ma'am is there. Yes.
Okay. It is now my great pleasure to introduce our final speaker for today's morning technical session. Dr. Najwa Mohamad, Associate Professor from the University of Cyberjaya, Malaysia. She is a distinguished pharmaceutical scientist, educator, and researcher serving as the Head of Quality, Head of the Department of Pharmaceutical Technology and Industry, and Treasurer of the Halal Center at the Faculty of Pharmacy, University of Cyberjaya. She earned her PhD in pharmaceutics from National University of Malaysia and has extensive academic and industrial experience, including serving as a product development manager in the pharmaceutical industry. Her research interests include drug delivery system, polysaccharide-based biomaterials, hydrogel wound dressings, pharmaceutical technology, cosmetic sciences, regenerative medicine, and halal pharmaceuticals. Dr. Mohamad has successfully secured competitive research grants, supervised doctoral researchers, and contributed significantly to pharmaceutical innovation and commercialization. We are delighted to welcome her to this International Faculty Development Program. So it is our privilege to have with us today. We sincerely thank you for joining us from Malaysia, ma'am. So the virtual platform is yours, ma'am, now.
Thank you for the invitations, Dr. Gauras Kumar and also Dr. Rubita. So now I would like to share my slide presentation, yeah? Okay, can you see the slide presentation? Is it okay, clear?
Yes, ma'am. Yes.
Okay. Okay, I believe this is a good afternoon, yeah, in India. So similar to Malaysia also, a very good afternoon to the MC and the host, and also for the invitation for me to talk about some of my research journey on the polysaccharide biopolymers. Okay. Okay, first of all, with the titles, okay, I introduce myself again. I'm Associate Professor Dr. Najwa Mohamad. I'm from Department of Pharmaceuticals, Technology and Industry from Faculty of Pharmacy, University of Cyberjaya. And this university actually very near to the airport in Malaysia. Is a few minutes only to the airport. And we are very near to our government city, which is Putrajaya. All right, so now I would like to present my research entitled translating biomaterials from polysaccharide to advanced therapeutics. So we use the engineered 3D polymeric networks for what application we use for the tissue regeneration, also for control drug delivery system. All right, so when we talk about polysaccharide, there are various of polysaccharide. And what is actually polysaccharide? Polysaccharide is a complex biopolymer that are with the beta 1,4-glycosidic linkage. And we can say that the best characteristic of polysaccharide as a biopolymer is non-toxic and is biodegradable and also is biocompatible. And where are they coming from? They are coming from microbial sources. They can be from plant sources and also from marine sources. So from the microbial sources, one of the example is bacteriocellulose. And from the plant source, one of example is actually we can say that is one of the example is Physalis has, and the example from the marine source can be from the alginates and also Chitosan. And from this biopolymer, the extraction of the polymers, we can actually fine tune to make into hydrogels, and this hydrogel can form into the film. It can form into the microneedles, control drug delivery system, and also tissue scaffold. So for this 1 hour presentations, I will actually tell you what is actually we experience to actually find to the polysaccharide into the various of applications such as wound healing, such as a control drug delivery system, and how we actually form hydrogen microneedles and also dissolvable film. Okay, so there are two things that we will talk a lot about today is the application of regenerative medicines because we would like to actually heal the wound that actually we can say that they have a severe wound, like let's say like a burn wound, okay, like the whole body, and we know that the skin is actually the largest organ that we have, right? And also the skin itself because due to the largest organ in our body, we need actually a large sizes, and also we need to accelerate the wound healing process because it's actually the skin barrier from all the, it's actually the first mechanism defense of our body. So we need to actually heal it as soon as possible when it's actually been open and also been injured, okay, and if there are any wound in the our first defense mechanism. Okay, so one of it is regenerative medicines, and the second one is controlled drug delivery system, right? And the controlled drug delivery system is we will actually to overlooks on the problem with the high dosage, and also the problem with inhalation itself is the frequent dosing like 3 times per day is like burden the patients. So sometimes we actually would like to formulate the dosage form maybe from a microneedles and control the hydrogel that can control the drug delivery. Maybe they can actually use it via the patch, or maybe for oral they can actually reduce from 2 times to 1 time. So it's actually to increase patient compliance. And also for regenerative medicine is due to the problem is due to the split screen graft. The problem is to get the even though it's a gold standard, but to get the healthy skin with the large amount is very difficult. Okay, so that's why we actually formulate the things that actually maybe we can enhance the productions by using the hydrogels. Okay, so the hydrogel is actually can solve, we can say that they have a potential to solve this problem, which is using the 3D hydrophilic polymeric networks of the hydrogels. Okay, so what kind of polysaccharide we have used in our research recently? We use 2 type of polymers, which is both of them is actually is from the food grade products, the one that we eat every single day. Okay, what kind of food? One of it is actually if you I can show here is this one the laser point. This one is nata de coco. All right. This is very famous in Malaysia and also Philippines. We eat it as a dessert. Okay, so nata de coco, and the other one is Physalim Has. I think this is very famous in India, and then also the food diet. I mean products. There's a lot of kind of mix up of fruits and also Physalim Has because they can actually swell a lot. All right, so when we talk about nata de coco, what we can actually obtain from nata de coco, what we can obtain from this dessert. Actually, we can extract the bacteriocellulose from this dessert. So where is this coming from? This cellulose coming from is from the fermentations of coconut water in the coconuts. They actually fermented with the sugar and they form a cellulose, and they form a cubicles structure like a nata de coco, and it been cut into the small and become a dessert to Malaysian population. So back then I use most of it. I use bacteriocellulose as my formulation of the hydrogels. And currently I moved to Physalim Has because they are more actually easier. We can say more easier during the extraction process. It can actually maybe if 1 month it took for the bacteriocellulose, And for the fisherman has maybe it took like a weeks. Okay, so it's reduced the time point for extraction of the cellulose itself. Okay, so this is two food grade that we actually use to form the hydrogels. So from the nata de coco, we call it as a bacteriocellulose, is a microbial cellulose, and also from the physalim husk, we not call it as a cellulose, it's a hemicellulose, is from the plant based, and also we use as for drug delivery and cell delivery. However, for the nata de coco that I will show here is only we focus on the cell delivery. We have other studies that we focus on drug delivery, but today I will just present on the one that we use for cell delivery. Okay, so as mentioned just now, the two polymers, that the main polymers that I use is from bacteriocellulose, from nata de coco, and also physalim husk. The one that the polymer that I extract is actually arabinoxylene polymers. Xylene polymers, and also the bacteriocellulose. So both of it is actually we can fine tune to make it a more the structure that how we want it. Okay, we can actually formulate the structure because it's a biopolymer that actually very good, that they have a very pure high solubility, and we can turn on the responsiveness due to different temperature, different acidics, and also is a we can say that is a smart controlled drug delivery system. Both of it can be used for regenerative medicine, cell delivery, and also controlled drug delivery system. Okay, other than like I mentioned just now on the wound healing, they can focus on tissue engineering, drug delivery, like microneedles. There are other industry that being used the foundations of arabinoxylene and bacteriocellulose, such as water purification in agriculture industry. So I think this is a biotechnology seminar. You can actually use it in a various we can say that category of industry. Not only we focus on the health formulation itself and target the health problem, and we will focus we will go to the first study first on the development of bacteriocellulose based hydrogel cell carrier containing keratinocyte cells and fibroblasts cells for open and burn wound healing. So first of all, we have the hydrogel. So what is this hydrogels? Hydrogel, we know that long ago it's been studied several times, we can say that several institutions already been studied the hydrogel widely, but is still remain the we can say that prominent candidate, best candidate to actually deliver it, and to one of it is a new studies, new venture to act as a cell carrier. So the hydrogel are 3D hydrophilics and cross-linked polymeric networks. They're actually capable to absorb large amount of water and is able to swell. And because of how actually this structure is because of they have like highly porous structure networks. That's why they are we are thinking of why don't we actually embedded the cells into these hydrogels. Why don't we actually formulate the tissue engineering, the scaffold to actually deliver the fibroblast cells that resembles the dermis layer, and also the keratinocyte cells that resemble the the epidermis layer. So the hydrogel are appealing as a scaffold because they resemble the natural soft tissue which include in the skin, and also the biocompatibility is very good. We can say that when we every time we actually analyze it through any cells, the toxicity is very compatible. There's no actually cause any toxicity to the cells that we use to study in our research. And it's non-toxic effects, and they have a lot of we can say that porous structure and good mechanical strength. And this application in the first study we use for the cell delivery to see whether it can improve the wound healing or not. Right, and as I said, the gold standard is actually require the healthy donor skin, and then it's actually cause extensive injury sites and severe pain, and also they have a high risk of infection. However, for the tissue injury skin substitute, we can know that it actually can increase the from a small scale to a larger scales. And it's non-invasive because it's topical application to the wound beds, and also it can actually actively, to stimulate the native soft tissue. And how did we produce the hydrogel? We use the green engineering, like one of it, because we have a lot of other methods to produce the hydrogels. One of it, we used to use electron beam irradiation. So we try to mix up the natural polymers, which is the biopolymer that from the food products like bacteriocellulose and also hemicellulose, which is the arabinoxylanes. And we grafted with the monomers, the acrylic acids, and we bombarded into the electron beam irradiations and to form the nicely formulated hydrogels. So what is the advantages? We not use any cross-linker, and also we can fine-tune the radiation dose to actually increase the porous structure or decrease the porous structure. It depend on the cross-link. It depend on the energy that we give, the dose, radiation dose to the hydrogel when we actually produce the hydrogel. So the previous study that we have published, we actually find out that they are able to heal the wound with only hydrogel alone, because they can actually absorb large amount of water, and also because of it can provide the water, moist and water to the wound bed. So we are thinking of why don't we actually embedded the active things inside the hydrogel. So we are thinking of why don't we deliver the original cells of skin to the wound bed, because they are actually rupture and they are we can say that they lost the epidermis and the also epidermis layer. Why don't we actually transfer the fibroblast cells and keratinocyte cells? So what we did actually, we harvest the skin cells. So I need to close my WhatsApp group, I think. Sorry for a moment, yeah.
Is there any issue, ma'am?
Is it okay?
Yes, ma'am.
Is it I'm sharing the slide?
Your slide
It's
is
not
displaying
working.
case A, constructing
Okay.
living dermal scaffold.
Okay, wait. I'm sharing again. Okay, now it's okay, right? Is it okay now?
Yes, ma'am.
Okay. So we harvest the skin cells from the original skin. The human skin, we harvest the skin cell from the we get that the skin from the we can say leftover surgery that actually been left in the hospitals during the surgery. So we take the leftover skin human cells, and we actually harvest and digest the skins and collect the keratinocyte cells and fibroblast cells and embed it into the hydrogels. So this is a method that we did. It's actually we construct the cell carrier and we harvest the cells and we construct the cell carrier, and we study on the cell attachment, cell migration, and cell transfer. And then we study in vivo to see whether it's able or not to heal the wound. So we measure the wound size, we stain with the H&E mesenteric and also immunohistochemistry. Proteins such as collagen, cytokine, alpha smooth muscles, actin, and involucrin. And this is the method that we, this is a powder that actually we get from the bacteriocellulose, and we mix up with the acrylic acid, and we use electron irradiation to form these gels. So this is how it look like, the hydrogels. And this is where we actually harvest the skin cells from the human skin. We harvest using the collagenous type 1s, and we get into the smaller small amounts, okay, and then we separate it with after that we grow in the media, and then we trypsinize it and then separate into the fibroblasts and keratinocyte cells. After we already separate the fibroblasts and keratinocyte cells, we actually construct into the cell carrier. So for the cell attachment, we actually embedded the keratinocyte cells, 3,000 cells, and the fibroblast cells, 3,000 cells of fibroblast cells. And we also studies on the cell migrations. We would like to see whether the cell can remain in that a similar place, or actually migrate into somewhere else, or immigrate into the deeper layer of the hydrogels, because we would like it to stay there, because you would like to transfer it to the wound bed area. And then after that, we also study on the cell transfer. So we use the control that resemble the extracellular matrix, which is the ovine collagen hydrogels. And we use the BCAA. This is the hydrogel that we use because of the name of bacteriocellulose and acrylic acid. So we name it as a BCAA. So you would like to see whether it's able to transfer or not into the ovine collagen hydrogels. Okay, so next. Okay, so this is a result that we will see that from the cell attachment up to 6 hours. The cell attachment is actually more than 70%. And this is where we actually see from the microscope where how actually the growing of the cells. We can see that the cell attachment is actually increasing, and similar that we see from this qualitative micrograph that actually resemble the quantitative numbers of the cells. And this is where we will see that how it's been transferred. This is from the bacteriocellulose, it's been transferred to the collagen. So we can say that cumulative that actually increase the percentage of transfer from the hydrogel to the collagen. And this is where we will see that whether they are actually migrate or not. So we can see that actually is kindly of like remain the similar we can say that stage, okay, up to day 3 to day 7. And we can see that it's actually most of it is been transferred to the ovine bovine collagen hydrogels. And this is overall of the result we will actually highlight. This we can see that it's the 80% mostly like 80% of fibroblasts and keratinocyte cells is successfully attached to hydrogel within 4 to 6 hours. So we can actually see that from the results. And also we can see that they are actually remain not that statics, but they are remain at the similar we can say that similar area from a 0 day to day 7. So it means that we can able to transfer it to the wound bed area. So when we actually study the in vivo, we actually studied in the burn wound nude mice, and also we studied on the open wound mice. We use the mice. So we see that up to 13 days, is actually 100% of the wound can actually close, but we can see that it's actually accelerate at day 7 with the cells. With the cell is actually accelerated from day 7, day 9, and also day 11 for the open wound. And also we did the meson trichome, and also we did the H&E staining. We can say that it's actually easily fastened to actually form a newly formed epidermis, and it will be actually confirmed back by the involucrin and CK14, when we see that this is a marker for the epidermal's growings. And also you would like to see that what is actually happening in the collagen level, which is increased or decreased in the dermis side. So we will actually see on the mesenteric and also alpha small muscle actin and also collagen levels of the under immunohistochemistry. And we will see that hydrogel cells, okay, is actually increase the dermal and epidermal regenerations, and also we can see that it's actively increase the myofibroblast activity. Right, so we can say that with the cells, it can actually increase the acceleration of the wound healing. So the key finding is actually we would like to see is hydrogel is actually able, right? This is the first and foremost that the study that we done. We would like to see whether it's able or not to deliver the cells and able or not to actually heals the wound. So we actually successfully to prove that yes, with the cells that the hydrogel can able to deliver the cells. And if you look at the porous structure of the cells, that is a very good, very good porous structure to actually embed the cells into the hydrophilic polymers. And this is an article that we published in the Drug Delivery and Translational Research with regard to the cell carrier, and in the scientific reports, and also in the one of the Elsevier book chapters with regard to the bacteriocellulose as a composite for the cell delivery. And the next project is that is the previous one that we use a bacteriocellulose. So after that, I've been explored to why do we use another type of cellulose and any other type of biopolymers, and then we use a similar electron irradiations, and then we would like to see whether it's able or not to form the hydrogel similar, the quality and the structure is similar to the bacteriocellulose. So we will try use the similar pattern, but we change the biopolymers. So we would like to study on what is happening in these hydrogels with different pH, whether it's responsive or not. Like a previous, like bacteriocellulose is very responsive toward changes of the pH and also changes on the temperature. Similarly, we use arabinoxylane. We studied on the, we can see that actually there's swellings occur so much in pH of 7.4. This is because of the carboxylic ion is formulate again that produced from the carboxylic acids. We can say that they are increasing the swellings, and when they actually increasing the swelling, they are capable to release the drugs. So one of the example that model drug, I think this one we use the paracetamol model drugs. For this hydrogel formulation, we use paracetamol to deliver the drugs. So this study is, we would like to study the development of this hydrogel via the arabinoxylanes, and we use the electron beam irradiations. And this is actually the water extraction of the Physalim husk. And we hypothesize that it can actually replace the conventional drug administrations. And also we combine the natural polymers and also the synthetic polymers and use the electron beam radiation, and we will see that whether it's able to or not to have the pH responsive, and actually whether it can actually able to deliver the hydrophilic drugs or hydrophobic drugs. And we also not using any cross linker while we are using the hydrogels, the formulation of the hydrogel via the electron beam irradiation. So this is the image of the hydrogel that we produce from the arabino xylens. And this is the characteristic that we actually studies. Okay. Okay, so for the swelling studies in the different pH, we can see that as seen in the graph, we can see that is a greater pH swelling in the pH of 7.4, but it will be shrink when they is actually in the stomach. So when it is in the stomach, they are not able to actually swell enough because they are a tendency to actually for COOH group to the group to ionize. So that's why it actually been swell enough in the pH 7.4. And we will see here at the room temperature, the swelling is not that higher when we actually put in the body temperature. When we actually put in the body temperature, it will swell more. And we can say that they are responsive toward the different pH and different temperature. Similar to what happened that we see in the bacteriocellulose polymers. And this is a pore structure that we see in the arabinoxylene polymers. It's kind of similar to the bacteriocellulose polymer as well. And we will actually see that there are a lot of pore size that can influence by the irradiation dose from the electron beam. So what happened is actually when we increase the dose ration, they can actually have the more packed and dense porous structure. And when we actually decrease the dose radiation, we can say that they are more large porous structure because they are more less dense. Cross-linking network between the monomers and the polymers. And this is where we actually loaded with the paracetamols, and we will actually go through the in vitro drug release studies. Okay, in the cumulative of pH, different pH, so we can see that in the pH of 7.4, it actually can increase the release compared to the stomach's pH. So overall, we will see that they have a micro architecture of the hydrogels, and the best ratio that we have here is actually 30-70 ratio of 30% of arabinoxylene and 70% of acrylic acids, and also the best dose is actually at 35 kilogray that we find out. And the swelling effect is more than 4,000% over 40 hours at pH 7.4. And drug loading efficacy is more than 90% drug loading efficiency toward the hydrogels. And the other studies, I mentioned that we also produce films from the arabinoxylates. So we actually call this is indicihils because they can be degradables in the fuel. It can actually be degradables. It's able to degrade. That's why it called invisible heat. So it can be used in the wound dressing. Okay, so we actually extract the arabinoxylates and we actually grafted it, all right, with the sodium arginate and also pectin, and we see that how it's actually the film is produced, and also we load it with the ganoderma. We load it with the ganoderma. We know that the ganoderma have the anti-inflammatory and also antimicrobial effects, and we observe that it's actually able to be dissolved and degrades. So here is a study that by using the pectin, and the other study also we use sodium arginate. So this is the grafted the characteristics that we studies on the porous structure of the films, and also we will confirm with the if there are the grafted of the arabinoxylates and also the pectin. And we studies on the antimicrobial. We will see that there are inhibitions of the bacteria in the Staphylococcus aureus and also E. coli. And the next one is we studies on the, I just share, okay, this is studies on the, what we did is actually we produce microneedles from the arabino-based hydrogels. So we actually would like to try different dosage form instead of we go with the orals. We try to use, maybe we can actually formulate the microneedles from the arabinoxylans and deliver the drone pazils. And the drone pazil is actually hydrophilic and is difficult to actually penetrate into the dermis layer. The epidermis itself, so we use microneedles, so it can actually rupture the epidermis layer to form a barrier, so the drugs can actually penetrate into the epidermis layer. So we use this one and hydrogel forming microneedle. We call it as a HFML, and we hypothesis that actually it can bypass the stratum corneum. And this is a very recent study that we studies using arabinoxylans and the others polymers, and then we characterize it. We haven't go through the in vivo part first. Maybe we will actually study the in vitro, then after that we will actually go through with the in vivo part. And this is the polymers that we use to extract from arabinoxylan, and this is a formulation we mix up with the solvent, using the solvent casting methods with the gelatin and also glycerol, and also we use the PVA, the percentage of PVA, and also the sodium arginate. And the characterization we studies on the morphology, the incision capability, the mechanical strength of microneedles, and we would like to study whether we are able to form the microneedles, how actually how long it how actually it can be insert into the parafilm, is it sharp enough? And then we also studies on the analysis of the FTIR, right, to see the functional group and compatibility of all the polymers in the microneedles. So here is the you can see that the methodology, we use the solvent casting method and we use a mold for the microneedles, and we actually have the reservoir of the drug itself. So the reservoir of the drugs, we use a polymers, and we mix up the drugs into the polymers and we put on top of the microneedles, and we actually see the permeations of drug release through the front cells. And we done the analysis to see whether there are a significant difference or not. So this is whereby there are process of the arabinoxylan extraction and purifications, and this is where they actually put the solution into the microneedles, then where we actually enhance the microcentrifuge, because there are a method that actually you can use the air pressure or you can use the centrifuge to actually increase the incisions of the solution into the microneedles. So this is a small that actually been produced, the microneedle that been produced. And this is a drug reservoir that where we actually loaded the drugs into this reservoir. We use 15% of PVA and also 50% of glycerol. And this is a microneedles that been produced. And this is whereby the morphology examination result, we will see that through the SEM. This is a shot of the microneedle that been produced. We can see that the height is around 390 to 425 micrometer, and also in between the interspacing between the microneedle is 300 micrometer. And this is we can say that actually it fall under ideal range, because the ideal range can be varied. It can be from 50 to 900s. So actually we can fine tune on the polymers, and also we can fine tune on the formulation to maybe to get it more sharper. So this is FTIR to actually see that where there are arabinoxylans and also identifiable peak in the FTIR spectrums. And this is the pH determination. We see that the pH is around 5 to 6 something, and they actually quite we can say that resemble the skin's pH. And when we put in the media, it's not actually cause any toxic effect to the cell itself. And this is a swelling where it actually depends on the polymer concentrations. So we can say that F6 have the lower swelling because the percentage of the polymer is higher compared to the rest of the formulations. And also we see that this is needle height reduction. So the idea is the microneedle cannot actually have the reduction lesser than 20%, but we see that F1 have the higher reduction due to because maybe it's too soft, it can actually easily to bend. So this is where we do the mechanical strength, and also we see that the compression before and after the compression. This is F1 before compression, and this is after the compressions. We said that highly bent. And we see that F3 is not that slightly bent after we actually compress with the 32 force under the texture analyzer machine. And this is where we observe into the microscope. Okay. And the insertion test is actually insertion test, we will see that what is actually happened. We try to use a parafilm layer, the first layer, second layer, and the third layer, and so fourth layer. We would like to see whether these actually microneedles are sharp to actually penetrate into the parafilm layer. So surprisingly, this is recently studies that we did, and this is kind of new things that we explore. We see that actually is the formulation, the hydrogel formulation actually is a bit sharp because it can able to penetrate into the third layer. But fourth layer is we can see that we are not able to penetrate for this current formulation. Maybe we will actually adjust the formulation and see whether it can penetrate to a deeper layer. So far, we can see that it can able to penetrate into first layer, second layer, and the third layer. And previous study, we can say that previous study that we see from the literature review, they are mentioning that the microneedle that they have is penetrate into the first, second, and the third layer. So we would like to adjust the polymers so we can actually increase the penetrations of the microneedles. And we also see whether it's able to or not to we have the drug reservoir, and whether the drug reservoir able or not to go through the microneedles. Yes, we actually see that it's actually able to release the drugs up to 24 hours. So now the patch can be used for 2 days patch. So it's able to actually release the drug, but there are so many things that need to be studied, especially on the in vivo part. This is just in vitro part. So the hypothesis actually from we can say that the hydrophilic drugs able to penetrate into the hydrogels, and when the hydrogel is actually swells, it able to actually release the drugs donepezil to the area of the epidermis, and it can actually penetrate into the dermis layer. And the cytotoxicity studies, we see that whether it causes any toxic effects to the skin cells. We can see that when we send the samples for the solubility, it's actually none of them is actually be considered as toxic because they have the solubility greater than 70%. And the last study that I would like to actually share is with regard to the design and fabrication and evaluation of arabinoxylene sodium arginate collagen these fibers biomaterial cross-link with the calcium chloride for tympanic membrane repair. So this is where I'm planning to actually use arabinoxylene. In the future studies, I would like to actually plan to use it as a cell carrier similar to bacteriocellulose polymers. So this is the first start for me to form the film first, and whether this film is suitable or not later to actually embed it with the stem cells to actually facilitate the wound healings and tympanic membrane. As you know that tympanic membrane perforation is actually mostly common for the younger age children. Because of this, tympanic perforation is actually impact their hearings, and also not only impact the hearing, they cannot actually swim in the swimming pool. They can actually have a normal life because they have the actually rupture and perforation at the tympanic membrane. So they are actually prone to get the infection. So when I read through the article, they mentioned that and also I find out that in Malaysia they only can have undergo the surgery for the tympanic membrane perforation up to when they are age of 12, 14 or 15, when they actually able to take care of their own ears. So for the younger age, when they have the problem with the otitis media, when they get infected, so they are difficult for them to actually get a normal life. They also have the impact of hearings. Okay, so the gold standard is donors surgery, but it will take so long. They will have to wait for the age become teenage. And also the others treatments, we can say that they are coming toward on the studies that they say that they would like to use the collagen biomaterials. But the problem with the collagen biomaterial is fast enzymatic degradation and poor mechanical property. That's why here that we would like to actually increase the mechanical strength of the collagen. So we actually embedded the arabinoxylenes, sodium arginate and collagens to form the films that actually able to transfer or deliver the cells at the tympanic membrane perforation. So this is where the methodology done is where the solution is being prepared and use the solvent casting, and then also we actually use the calcium chloride as a cross linker. And we use the phase drying machine because the collagen is very sensitive to we can say very sensitive to the temperature. So this is where we actually form arabinoxylene, salt arginate, and also collagen biomaterials, and we see on the thickness whether it's suitable or not, the thickness of the membrane to be delivered the cells to tympanic membrane perforation area. And we see that the pH whether it's suitable or not, because it's to insert into the inner layer. And also we see that whether it can actually swells, and also how is the porous structure, and also how actually the polymers that grafted, whether the collagen is still there, the sodium arginate and arabinoxylene is still there in that biomaterial, and we see on the toxicity of the cells. So this is a results where we actually form films that from these formulations, various formulation from F1 to F7. We will see that they actually successfully form a formulation using the solvent casting method, and all is intact, and we can see that they have a very good stability and structural and integrity that actually able to, one of the candidate can be able to actually deliver itself. So when we see the thickness of the biomaterial is a range around 0.105 to 0.17 mm. So we see that when we actually increase the concentration of the polymer, the thickness of the biomaterial also will be increased. And we will see that when we actually see that we see the citation, what is actually the best best thickness that should be used for the tympanic membranes. They mentions that according to Jia et al., the par tensor is around 109.2 to 135 micrometer. And we can say that par placida is actually considerably thicker, ranging from 78 to 300 micrometer. And all formulation is in between the, we can say that it can be considered in between the what is being suggested via the Jiang et al. So we see that, oh, that is maybe the thickness actually quite okay. So we further find out that what is actually the pH. The pH is around 6.9 to 7.477. So there's no adjustment on the acidic molecules or alkaline molecules. So we can actually mostly is a very close to physiological pH range. And if it's very close, it's actually we will say that is not cause any toxicity to the skin fibroblasts. And this is a water uptake and also the swelling ratio. We can say that they are actually more than 3,000%. It can actually swell. It resembles the hydrogel just now. And we can say with the collagen and sodium arginate, so they can remain the high percentage of swelling. So, so this is actually been proved that actually they are a pore structure inside the films. So we can say that all the formulation exhibit a very high swelling is more than 2,000%, indicating suitability for tympanic membrane repair by supporting the flexible and supporting the hydration and also nutrient diffusions for the tympanic membrane perforation repair. And with regard to in vitro biodegradation studies, we can say that it's highly biodegradables. The film is highly biodegradables. So we find out that it's only able to be transferred. Okay, the film can be used to transfer the cell, but not yet to actually stay within the membrane. So if you would like to actually increase the time, requirement for the film to be stay there in the tympanic membrane. So we actually maybe have to fine tune the polymers to increase the biodegradation rates. Actually, we can fine tune to increase more the polymers so it can actually stay for 4 weeks times. So for the time being, we can say that it's able to only transfer because the biodegradation is very rapid. And we can say that easily lost. So there are pros and cons on the films. So this is a structure of the polymers that the film structure of the polymers that we were planning to actually embed the cells, the stem cells, to actually deliver in the tympanic membranes perforations. Right. And this is FDIR that actually we will confirm that there are possible of the amide groups from the collagens and amide 1 and amide 3 groups from the collagen, and also it resembles the arabinoxylane structure. So this is arabinoxylane structure, and this is where they are grafted with the film that contain the sodium arginate and also the collagen. And the results here, to see this is a last result I would like to share. This is where we actually see on the biocompatibility of these films. Actually, it show that more than 70% of the cell viability is indicate that it's not toxic and also is biocapable. We can say that with the low percentage is almost more than 100% of the cells actually viable during the MTTAC. So overall, that is actually the the studies that I shared through the various of the polymers that being used to actually for the various of application. We can use for cell delivery application, for drug delivery application, and also we use kind of the waste products of the food product that we can actually cycle into the clinical interventions, and also sometimes that we use electron irradiation, we can avoid to use any chemical cross-linker, and we can remove all the unwanted monomers, and we can actually easily to reduce the unreacted monomers during the radiations. And maybe we would like to explore more in the polymer matrix in the future for different uses, and also the clinical challenges that we will actually able to see in the future. I think that's all from me with regard to the one that I'm sharing. So I give back the floor to the MC.
Thank you very much, Dr. Najwa Mohamad, for your insightful talk. And this presentation was a remarkable insight into the development of translational and potential for this polymeric biomaterials.
So much.
So participants can ask the question in the chat box, can post in the chat box or raise their hands.
Hello, Dr. Najwa.
Yes.
Yes. Ma'am, I had one question that once you have created that film with this biomaterial, I want to know if it is breathable, like does it absorb further or not?
Okay, can you refine again the questions about the films?
I have the question I have is that is the biomaterial that you have created, the film, the say bandage, whatever you have created, that is it breathable? Is it further absorbed stuff?
Yes, for the hydrogels that I mentioned just now, for the hydrogels, this actually it can be used for the wound dressing itself. It can actually absorb a large amount of water.
So the film is, that is also hydrogel.
Okay, there are two the one that I share, one is the films and one is the hydrogels. So the one with the hydrogels.
I'm talking about films.
Oh, the films with the arabinoxylene film, is it?
Yes, yes.
Okay, that one is for the tympanic membrane. No, it's only for the transfer the cell carrier, and also as a cell carrier only.
Okay.
It's not for the wound bed. For the hydrogels, yes, it can act as a wound dressings, and also it can act as a cell carrier together. But the films, the tympanics, for the tympanic membranes perforation, no. We only want it to be able to deliver the cell keratinocyte cells and fibroblasts, or maybe the stem cells, and we want them to actually degrade only as a cell carrier.
Okay, okay. Thank you. It was a great talk.
Thank you so much, Dr. Gupta.
So, as a token of heartfelt appreciation and gratitude, we are delighted to present a digital memento and certificate of appreciation. May I request Dr. Gaurav Kumar to kindly present the memento? Thank you, sir.
Thank you so much, ma'am. On behalf of Department of Biotechnology and Bioengineering, Galgoti University, we proudly present this digital memento in recognition of your outstanding contribution to this faculty development program. And we are honored to present this e-memento. So kindly accept it.
Thank you so much.
Thank you so much,
Thank you
ma'am.
so much for the invitation. Yeah.
Thank you so much, ma'am.
So that concludes our morning technical session for day 2. And we extend our sincere gratitude to all our distinguished speakers and the participants for this engaging discussions. And we look forward to welcoming you to the afternoon session as per the program schedule. So thank you and have a pleasant break. So we are expecting you to join at 1:30. Thank you, everyone. So good afternoon, everyone, after the lunch. And on behalf of Department of Biotechnology and Bioengineering, School of Biosciences and Technology, Galgotias University, I warmly welcome you back to the afternoon technical session of day 2. So without further delay, let us begin our next technical session. It is my great pleasure to introduce our next distinguished speaker, Dr. Marieswaran, Assistant Professor in the Department of Biomedical Engineering at the National Institute of Technology, Raipur. Dr. Marieswaran earned his PhD in Experimental Biomechanics from the Indian Institute of Technology, Delhi, along with an MTech in Biomedical Engineering from VIT University, and a bachelor's degree in biomedical engineering from Anna University. His research interests encompass experimental and orthopedic biomechanics, medical instrumentation, biosensors, biomaterials, computational modeling, biomemes, biomicrofluidics, medical imaging, biophotonics, and FDM-based 3D printing technologies. His work focused on developing innovative biomedical devices and assistive technologies that bridge engineering principles with clinical applications. Dr. Marieswaran has published numerous high-quality research articles, book chapters, and conference papers. He has also actively organized international conferences and self-sponsored workshops while mentoring students and researchers in cutting-edge biomedical engineering. So we are delighted to welcome Dr. Marieswaran to this International Faculty Development Program. On behalf of Galgotias University and the organizing committee, we sincerely thank you for joining us today. And it is indeed a privilege to have an expert with your outstanding academic and research background, sir. So the virtual platform is yours, sir.
Thank you. Thank you for introduction, and thank you for inviting me to be part of this FDP. So I think I can share the screen. Is my screen visible?
Yes, sir. Yes, sir.
Yeah. Yeah. So good afternoon, everyone. I want this session to be interactive. Can stop me then and there and ask questions, and I will be more happy to answer. So this today's talk I have designed in such a way that I will be introducing the concepts of polymer additive manufacturing, which is nothing but 3D printing. And then we will see some medical applications, and then we will go into bioprinting. I don't want to directly go into bioprinting. So we will see some basic concepts of additive manufacturing first. So you will see what is additive manufacturing. Additive manufacturing is nothing but you are going to fabricate a part layer by layer. So you are going to add materials. So this is different from the conventional manufacturing technique, which is called as subtractive manufacturing. Okay. So we will see what are the major components of additive manufacturing, what are the steps involved in additive manufacturing, and then we will focus more on polymer additive manufacturing. Okay. And then we will see what are the practical issues when you start 3D printing, what problems you might face, and then what kind of solutions are there, and then what are the steps involved in printing, post-processing. Okay, what are all the materials involved, from where we get the materials. Okay, and then we will we'll also see what are the pros and cons of particular 3D printing technique. Here we are focusing on the polymer additive manufacturing. Under polymer also we are focusing on fused deposition modeling, and then applications of FDM in medical field we will see, and then later we will go to bioprint. So let us. Yeah. So what is additive manufacturing? As I told you earlier, so the right side, what you see is additive manufacturing. So you are adding material layer by layer. Okay. And here we have in the left side, we have subtractive manufacturing. This is a conventional or traditional manufacturing method which is followed in mechanical and automobile industry. Okay. So you take a big block of material and using a drilling or milling tool, you remove the unwanted parts so that you get the actual part of interest. Here you are adding material layer by layer. So this is also a top-down approach. This is a bottom-up approach. Okay. And then here the wastage is less, here the wastage is more. In additive manufacturing, you can have complex designs can be easily fabricated compared to subtractive manufacturing. Okay. So we can have different layer heights. We can start as small as like 20 micron, 50 micron, and we can go up to 1 mm, 3 mm. So that is the thickness of each layer, which is going to be deposited on top of each other. Okay. That is the kind of definition of additive manufacturing. Okay. So it allows physical components to be made from virtual 3D models by building the component layer upon layer until the part is complete. So that is the process of additive manufacturing. Okay. So we have conventional tools like CNC milling, okay, which is used in subtractive manufacturing. In additive manufacturing, we have a lot of techniques. We will see them. I will give an introduction of all, and then we will focus on more on polymer additive manufacturing techniques. Okay, so these are all the steps involved in additive manufacturing. You have a design which is nothing but a 3D model, virtual 3D model of the part which you want to fabricate. So that design you will be doing in any CAD/CAM software, like SolidWorks or Fusion 360. So once the design is ready, okay, you import the design in the software, in the 3D printing software. You export in STL form and import that STL file in the 3D printing software. Okay. And then in the software of the 3D printer, you slice it. What do you mean by slicing? You take the entire object and then you divided it into horizontal layers. So that process is called slicing. Okay. Imagine you have a 10 mm object and then you are slicing it at 1 mm height. So you have 10 slices. Okay. More the number of slices, more accurate you are going to fabricate the prototype. Okay. So the slicing part is done in the printer software and then you do the actual 3D printing. Okay. The commands are given to the printer to deposit the material layer by layer, where to deposit in the XY coordinates. Okay. And what are the other parameters, like what is the temperature of the material, and then what is whether you need a support or you don't need a support. Okay, so these things are commands are given to the printer sequentially. Okay, so and the product is 3D printed, and then finally you do post processing. In post processing, what you do first, you remove the part from the printer bed. Okay, and then you do some post processing like surface finishing, improving the surface quality. Okay, sometimes there may be some support materials which you have to remove it. Okay, so we cannot build subjects which is having overhangs. Any structure which is having more than 45-degree with respect to the horizontal layer, we cannot print it without having a support structure. So those support structures has to be removed. And then wherever the support structure is touching the object, the surface quality is bad. You have to improve the surface quality by doing some surface finishing. And there are some particular 3D printing techniques which uses UV light to cure. So in those cases, again, you have to use UV light to cure the residual polymer. Okay. And then sometimes in metal additive manufacturing, you have to put it in a high temperature furnace for all the residual stress to be removed. So those things comes under post-processing. Okay. So these are all the steps involved in 3D printing, right from design, slicing, printing, and then post-processing. So we can also classify the different 3D printing techniques based on the raw materials with which we print. Okay. So if you are starting with a polymer, okay, and the polymer is a thermoplastic polymer. So the thermoplastic polymer means it is when you heat the polymer, it liquefies, and when you cool it, it solidifies. So it is dependent on the temperature. It has a temperature above which, when you heat the polymer, it will become semi-solid, and then when you cool it back, it becomes a solid. So that is the basic concept of fused deposition modeling. Thermoplastic polymers are used. In some cases, you heat the polymers directly using a normal heating element. In some cases, you use laser to deliver the heat to the polymer. So when the laser is used to deliver the heat to the polymer molecules, so it is called a selective laser sintering. In selective laser sintering, the raw materials are in powder form. Okay. And in fused deposition models, you have the raw material, the starting point is a filament. Okay. You can have pellets also. Nowadays, we have printers which work on the concept like directly from the pellets, you can 3D print. Ideally, from pellets, you make filaments. Those filaments are used to 3D print. That's a two-step process. But nowadays, there are some printers which are equipped, which can directly print from the pellet form. Okay. So when the raw material is in the pellet form or in the filament form, so the thermoplastic material, the printing process is called as FDM, fused deposition modeling. And then we have thermoset polymers. Okay, they are nothing but liquid resins. So here the starting point is the liquid resin, which are sensitive to UV light. Okay, ultraviolet light of particular wavelength. So they have photo initiator, which is sensitive to a particular wavelength. So these photo initiators are activated when UV light is given. So when those parts become harder, where the part which is not exposed to the UV light, they remain softer and they are washed away in the development process. Okay. So again, we have digital light processing and stereolithography, different printing techniques, depending upon how you project the UV light on the polymer. So these are all for polymers, solid polymers, liquid polymers, powder polymers, filament polymers. And then when you have metals as raw material, that's a starting point, we can have metal powders. So the metal powders, if you you can use laser to deliver energy to the metal powder, and it will melt the metal powders. It is the selective laser melting. Okay. Again, we have electron beam melting also, where again fine powders of metals are heated using the electron beam instead of the laser beam, and then you end up with a 3D printed object. So layer by layer, the metal powders are deposited, and then you have electron beam, which is focused on the layer, each point in the layer. So the electron beam delivers heat, and then it completely fuses the two molecules of the metals. Okay, and then you can have laminated object manufacturing also, where you can have layers of paper or plastic or metals which are stacked upon each other. So instead of adding each point, we will add a layer at one go, and then we will try to attach this layer to the layer which is beneath. So that is the laminated object manufacturing. Here we can use metal layers also. We can use aluminum sheets. Okay, and then we can do ultrasonic, that is called as ultrasonic additive manufacturing. So this is the general overview of the different or classification of additive manufacturing techniques based on the raw materials, whether it's a metal polymer or whether it is in what is the form of the raw material, whether it's in the liquid form or in the powder form or in the filament form. Okay, so in this lecture, we will be mostly focusing on the FDM 3D printing techniques. It is the most cheapest technique, and then it is easy to use. Technically, it is not very complex system. And then we have it is available from 15,000. You can buy it from Amazon also. Okay. So here you have as I explained already, you have a thermoplastic polymer. So that is in the form of a filament. So that is pushed through a nozzle. The nozzle is heated to the melting temperature of the filament, and then the nozzle moves on XY direction and it prints the material. Okay. So the build platform then moves down a fraction of millimeter, and the next slice is deposited on the top of the first slice. So again, it's a repetitive process. So it deposits material on one layer in XY coordinate dimension, and then the bed moves down, and then again the next layer is deposited. So this is called as extrusion, material extrusion or filament extrusion system. Okay. So we can have support structures. Okay, and the support structures can be either removed manually or you can use some chemicals to remove it. Okay. So this is the system. So you have a build plate, and then the material is deposited layer by layer. Okay. So the black one is the support material. See, you can see this structure is like a cantilever. Cantilever is, or it's some structure which is supported on one side, but it is not supported on this side. So imagine this is like a horizontal plate which is being supported in one side, but here there is no support. So this is called as overhang. Okay, so this overhang cannot be printed unless you use a support material. So these darker areas are the support material. So we can have different nozzles to print, one to print the actual structural element and one to print the support element. Okay, and these support elements can be removed later, either physically or you can do some solvent treatment to remove it. Okay, and then it deposits each material layer by layer. Okay, so this is the filament which is put it into the nozzle. In the nozzle, you heat the nozzle so that the filament reaches the melting point and it comes out in liquid form. Okay. So this is the step-by-step preparation. You have the design. For example, you want to print a sphere. A sphere is, so you imported it in STL form and then slice it in the 3D printing software, which is nothing but you make horizontal layers or cuts of the object. And then each horizontal layer is 3D printed layer upon layer. Okay. So this is somewhat at intermediate state what it will look like. And then you give other parameters also while doing the slicing. You give layer height, printing speed, retraction speed, infill type. You can print a hollow sphere. You can print a partially hollow sphere, 50% hollow sphere. So these things we can play not only in the design, but also while 3D printing. Okay. You can have a sphere which is having another material inside, and the superficial coating is made up of a different material, different polymer. So that is also possible. Okay. So these parameters you give it in the software. So am I connected? Hello? Hello?
Yes, sir. Yes, sir. Yes, sir.
Yeah. Please. I just wanted to check. So this is the, there are some gears which are going to push the filament into the nozzle. So the nozzle is the hot end. Okay, so we have different kind of systems where the gear is placed away from the nozzle hot end and where the gear is placed just above the nozzle. So that is called as direct extrusion system. And when the gears are placed away from the nozzle, it is the Bowden tube system. Okay, so the gears move in clockwise or anti-clockwise direction, and they propel the filament in the forward direction. Okay, so this filament is pushed forward into the nozzle, which is hot, by the movement of these gears. Okay, but again, positioning of the gears will give different advantages and disadvantages. Okay, So when you have a direct through extrusion system, so the printing speed is less because the nozzle is moving in XY direction and the gears are sitting on the top of the nozzle. So the weight of the moving part is high. When the weight of the moving part is high, right and not moving. Yeah. So this is the moving part. So the filament, the gears are sitting on the top of the moving part. So the entire part has to move. So when the entire part has to move, the weight of the part is higher. So you end up the compromising with the speed. Okay, the system cannot move at higher speed. Okay. So that is the problem with direct extrusion system. The speed is low compared to Bowden, but the print quality is very good in type of direct extrusion printers, and then it supports flexible filaments also. So the filaments which are used in FDM 3D printing, they are not only brittle. Some we can use some flexible filaments also. Okay, we can, but for using flexible filaments, TPU is a flexible filament. Okay, transparent polyurethane. So you should have direct extrusion setup. Okay, so this is the difference between the powder and direct extrusion. The hot end nozzle is here and the gear mechanism is far away. So when it is pushing a filament like PLA, which is brittle, polylactic acid, it's a brittle. Hello.
Yes, sir. Yes, sir.
Yeah, I got disconnected in between.
Yes, sir. Yes, sir.
Yeah, I think. Yeah. So which slide were we talking before it got disconnected?
Sir, extruder you are showing.
This one?
No,
So extruder of
Extruder.
PLA, yeah.
This slide.
Materials.
Sir, slide is not visible.
Okay, I have to share the screen again.
Yes, sir. Yes, sir.
Yeah, this
Yes.
slide.
Yes, sir. This one only.
Yeah. So you also please talk to me in between so that I know that I am connected. Okay. So yeah, this is the setup where it shows different placement of the gear mechanism. Okay, whether it is far away from the hot end or the nozzle. Okay, so in that case, brittle filaments, they are pushed in synchronous. Okay, the filaments are pushing and they are coming out of the nozzle without any delay. When we use the Bowden extruder, in which the extruder is, the gears are far away from the nozzle. But when you use a flexible filament, so you can see in the side, left side, that structure is printed using a flexible material. So only the structure is also very flexible. So that is printed with some flexible material like TPU. So when you use a flexible filament, when a gear is pushing the filament in the Bowden assembly, it will not directly come out of the nozzle. It will undergo some bulge or kinking in the in-between place, and then it will come out with a delay. Okay. So this delay will introduce some problems in the final print. Okay. So it's better to move this gear system near the hot end as is shown in the direct system. Okay. Where the delay is there, but still the delay is reduced. There is a smaller delay. Okay. So when you use a flexible filament, okay, not only the pushing the filament forward is important, you have to push the filament out of the nozzle also. So when you do 3D printing, okay, it deposits material at some point and then it will travel over some region where it doesn't deposit. In that places, the filament has to be retracted. So it is always pushing the filament forward and taking it back. So that is called retraction. So the retraction is a bigger problem when you use a Bowden setup and then when you use flexible filaments. Retraction is okay when you use a PLA. PLA is a brittle filament. But when you use a flexible filament, okay, retraction, there is a big delay. That means you end up depositing material where you are not intended to deposit material. Okay, so again in direct setup, this delay is reduced. Okay, so these are all some problems which is because of problem with the retraction. Okay, this is a small structure. Okay, you can see some spider web kind of material between the pillars. So that is called oozing. The material is oozing out of the nozzle when it is traveling. Okay, ideally it will print material here and it has to travel this much distance without printing and then again it will print. So when it is traveling without printing, the filament has to be retracted out. So that retraction is possible only when you use especially when you use a flexible material. Okay, the retraction will introduce a lot of delay when you use a Bowden extrude. Okay, the right hand side you can see this spider web kind of anomaly is not there because we have given the correct retraction setting to the printer. Okay. So again, warping is another problem when you do FDM 3D printing. Okay, there is the heat distribution is not uniform. Okay, the first layer which is getting attached to the bed. Okay, when the first layer is printed, it will stick very nicely. But as the layer printing is going continuously, the material is deposited at upper layers. The lower layer or the first layer started cooling. When the cooling is not uniform, okay. So you are seeing a 2D image. Ideally, it's a 2D slice. In the front view, you are seeing ideally it is a rectangular profile. So the rectangular profile, every spot has to cool in a uniform manner. So if the cooling is not in uniform manner, some part will detach from the bed. So that is called as warping. You can see here in the left side. So one corner of the rectangular slab has detached from the bed because the cooling is not uniform. Because of non-uniform cooling, the forces are not equal. So the forces are higher, sinking forces are higher in one place and it pulls the object from the bed. So it leads to detachment. Okay. So this is called warping. So these warping and oozing, these are all problems in FDM, which you can control by playing with different 3D printing settings. So this was done in my lab. So we have dual extruder. So at the same time, we can use two thermoplastic materials to print one object. Okay. So I had printed a vertebra with these things are support. The red one are the support material. Okay. So the support materials can be removed. They are mechanically not that stronger. So this is a part, the third image is a part of a dice. Okay, it is actually a single part which is having two subparts. So we can print the two subparts with two different material. So this just I wanted to show that we can have multi-material printing. Okay, and then these are the stations which are used to remove the support materials. There's a pressurized wash chamber where you can use high pressure water jetting to remove the support material. Okay. And then Cura is the basic slicing software. So I will show some steps in Cura. Once the design is made, okay, you import the object in the design software and then you give the printer specifications. Okay, what is the bed size of the printer, XYZ dimensions of the printer, and what is the how many number of nozzles are there in the printer. So Ultimaker is again a very common printer. So this is just a dog bone design which we imported in the sliding software. And these are all the different parameters you can give to the object while printing. Okay. And then what should be the infill, how many layers should be there in the bottom, what is the speed, what is the material. So you can select different infills. Even some TPMS structures can be used, some periodic structures can be given. And then what is the material? Okay, what is the heating temperature, melting temperature of the material, density of the material? So every parameters you can give in the slicing software. Okay. So again, from where we will get the material? As I told you, FDM works with filament. Okay. So how we get the filament? So ideally we get the extrude the filament from the pellets of the polymer. For that we have another one setup called as filament extruder. Okay. The filament extruder is nothing but we have a hopper and then we put the polymer pellets in the hopper and then it is processed to different heating zone through a screw and finally it comes out of the die, circular die, which is giving us the filament. So you can extrude filaments of different thickness from the polymer pellet using this extrusion machine. And then you can have not only polymer filaments, you can have composite filaments. You can mix some other polymer or some other material with the filaments. So people are working in lot of research areas, especially making use of these concepts. Okay, people are working on drug-eluting implants. People are working on creating polymers which are conductive. So you can add a conductive element to the polymer and make it conductive, or you can add some reinforcements to the polymer, which will make the polymer even to exhibit better mechanical properties. Okay, so there are different applications of adding another material in the polymer matrix. So what is a composite? Composite is nothing but you have mixture of two materials. Okay, you have a matrix in which another material is introduced. Okay, so generally we are talking about polymer matrix in which you put a metal or you put another ceramic or another amorphous form, and then you end up with some composite filament, which is going to have a combination of both the polymer also and the infill reinforcements also. Okay, generally people introduce composites to increase the mechanical strength or to improve the thermal performance. Okay, you want to put the printed object in some high temperature environment or high pressure environment. So you want the thermal performance or mechanical performance to be better, or the electronic performance. You want to use it as a sensing element. You can do some wearable electronics, wearable sensors can be 3D printed if you are able to make the polymer conductive by adding some elements which is making it conductive. Okay, we can have, like I told you, drug-eluting polymers are also fabricated using this method. So this is the machine which is used for extrusion, and then FDM 3D printing has its own problems also. It is not, as I told you, it is very cheap, but it has some. The surface finish is poor compared to all other 3D printing techniques. If this is the CAD design which you wanted to fabricate, the actual print will be something like this. So this is called the staircase effect, because you are adding layer by layer. Okay. So the staircase effect is more prominent if you have more curvatures or more bends in your design. Like this is the design. So this will be the actual print. This can be addressed by reducing the layer height. But again, FDM, you cannot reduce the layer height less than 150 micron or 100 micron. So that is the challenge of this technology. We cannot go below 150 micron or 100 micron. So there are other fabrication techniques which can go up to 30 micron. Okay, those technologies, this problem will be, the staircase effect will be reduced. Or we can do is, after 3D printing, we can do some surface treatment. We can do some rubbing with sandpaper so that the finish, the staircase effects is reduced. The finish is improved. The surface finish is improved. Okay. So these are all the different materials which is used. Commonly, we use ABS, PLA, nylon, then TPU is a flexible filament. PEEK is getting more attention nowadays, medical attention. So PEEK is all the other polymers. They are like low temperature polymers. Like you can print it between 190 to 250 degrees Celsius. For PEEK, you have to go more than 400 degrees Celsius. Okay. So PEEK is a chemical compound which is used in a lot of aerospace applications also and medical applications also. It is polyether ketone. Okay, it is having higher comparable property to the mechanical property of the bone, but it is weaker than titanium. Okay, but it is the in the case of better performance to avoid stress concentration, PEEK is more suitable compared to the metal implants. Okay, and we can have metal filled polymer filament also. Like as I told you, we can make composites made of composites where the polymer is the matrix and you can have metal filled in between. So these materials can be used. Either you should have them in pellet form or in the filament form. So the different advantages and disadvantages are there for the FDM 3D printing techniques. So it is, the advantages is the most affordable. It is printing technique. It is not considerable for manufacturing. For proof of concept, for research, it is more suitable. Okay, it prints in standard engineering thermoplastics. It is the raw material is also cheaper, the filament. Okay, and the equipment is also cheaper, and it is easy to operate the machines. So the disadvantages of this method is it is the most anisotropy process when compared to other fabrication techniques. Okay, because you are adding layers in the z-axis, so there is substantial weakness in the z-axis. So the material, when you pull the material from the top direction, so the layers are going to get detached from one by one. So the material is weakest in the z-axis when you load it in the z-axis. Okay, so that is called as anisotropy. So the final product is not having an isotropic behavior. Isotropic means it is going to exhibit same mechanical behavior when you load it in any direction. I would give the simple example as a smiley ball. Smiley ball is where in whichever direction you are going to press the ball, it will get shrink in the same way. But not all engineered structures are like smiley balls. In some directions, they are more stronger, and in some directions, they are weaker. So that is the anisotropy in the weakness in the z-axis. And then we have poorest surface quality. It requires support materials for overhangs. So this problem is there for other 3D printing methods also. And then if you have a support material which is deep within the structure, it is difficult to remove it also. So we have to use some chemical agents to remove the structures, support structures which are difficult to remove. Okay. And then we can also, this is one applications using FDM where they have made a surgical jig. So the surgical jig is 3D printed and then it is used intraoperatively. So that surgical jig is 3D printed by FDM 3D printing technique. So it is useful for, this jig is used for putting the pedicle screw directly within the pedicle. Okay, this is a spine surgical process where you put a screw within the pedicle region. Okay, so it is very delicate because if you are not exactly placing the screw in this region, you may damage the neural canal and the patient can become quadriplegic or paraplegic. Okay, so you should exactly put the screw in this direction. You cannot go within and spoil the spinal canal. Okay, and if you come out in this side, the screws will be looser. So it will be not having a good fixation. It will be loose. Over a period of time, the screws will come out. So to put the screw exactly in this direction, we are using a guide, surgical guide. That surgical guide is 3D printed. Okay, so FDM can be used for that process. So this will reduce the time the surgeon will do for insertion of the pedicle screw. Okay, and then we have peak 3D printing. So this is a peak implant, which can be used as an intervertebral disc replacement. When the intervertebral disc is damaged, you remove it and you put cages. So these cages can be of titanium. Generally, they are used of titanium, but PEEK is now coming up. Instead of titanium, we can use PEEK. And then it will immobilize these two levels of vertebra and it will fix them. Okay. So PEEK is coming up, catching up with titanium, especially for intervertebral disc replacement surgeries or procedures. And then we have other 3D printed structures using DLP, in which you use them you print them for making phantoms, which are used for different radiological Q&A applications, quality assurance applications, where you want to mimic the attenuation behavior. Okay, we cannot do calibration with the real human. Whenever you have a CT machine or X-ray machine, they do repeated calibration protocols and then they use phantoms instead of the real humans. So the phantoms should have comparable attenuation coefficients to the radiation energy, similar to what the human tissues exhibit. So we have different materials, okay, which are offering the attenuation behavior similar to human tissues. Okay, here we have a 3D printed spine phantom. Okay, and then we have different thoracic and lumbar vertebrae, and then they are able to model cancer tissue behavior also. Okay. And then, obviously we have orthopedic implants. Nowadays we have 3D printed orthopedic implants, which comes as part of the metal printing. So with this, I complete the general overview of the 3D printing techniques. We will go to the bioprinting aspect. Okay, as I was told, so most of the audience are from biotechnology background also. So I thought I will be touching up the bioprinting aspects also. So bioprinting is coming up nowadays. We can directly mix the tissues, okay, cells, and the factors, growth factors, scaffold, and then we can mix everything, and then we can make a 3D structure. And then that 3D structure can be put in a reactor for maturation, and then the organ or the tissue is ready for transplantation. But why we have to do this? Okay, so that will tell us what is the advantages of 3D bioprinting. Okay, what is the need for bioprinting? What are the problems in 2D cell culture and animal models? That we will go through first. And then we will see about the in vitro 3D models. And then what are all the advancements in the microfluidic organ on chip domain? What are all the 3D bioprinting techniques? From where you get the components for 3D bioprinting? From where you get the cells? From where you get the extracellular matrix? Should we use synthetic polymer or natural polymers? Should we use stem cells as stem sources? And what is the role of bioreactor? As I told you, bioreactor is the place where the tissue which is already made by the 3D printer is given some external stimuli, either in the form of mechanical stimuli or some shear stress is applied, some electrical stimuli is given. Okay. So depending upon the end use of the tissue, for example, you are making a blood vessel. So blood vessel have a pulsatile, the vessel wall, they are pulsatile in nature because of the pumping activity of the heart. So you have to subject the developed vascular model to the pulsatile flow for maturation. Similarly, if you are working on some neuronal application, so you have to give electrical stimuli to the cells which are 3D printed, and then you end up maturation. Okay, and then you can have different other stimulus also, and finally we will see different extrusion based or inkjet based printing techniques. Okay, what is the need for bioprinting when we have already 2D cell culture? The 2D cell culture studies, the cells are grown in 2D condition and they are substantially different in their morphology, and the cell to cell interaction and cell to matrix interaction is different when compared to the cells which are grown in 3D environment. Here we have a flask in which the cells are grown. Okay, you have a 2D cell culture. The cells are attached to the layer surface of the cell culture flask. Okay, and it is having minimal interaction with the medium and with the other cells also. Especially when you have a 3D environment, you have a 3D printed cell or 3D printed tissue. So it is the interaction with the extracellular matrix is different because of the three-dimensional availability of the ECM to the cell. Okay, so that is one major advantage. And why we can't use animal models? Why we should use bioprinted models? Generally, we study mostly in animal models some disease behavior, response to the drugs. But the problem is sometimes the system is so different. The drug which works for animal may not work for human. The drug which may not work for animal may work for human. So it is difficult to model autoimmune diseases, cancer, to induce cancers in animal models. So it is better to use a human cell derived bioprinted setup for these kind of applications. For example, you have invented a new drug. You want to you want to test how the drug is effective. So it is for blood vessel or it is for some nephron. So you want to simulate that blood vessel or nephron and give the drug and see what are all the changes the drug is causing in the neuron or in the blood vessel. So we cannot directly go to clinical study in live humans. So we have to do this in some lab simulated models, which is similar to live humans. So there the bio 3D printing gives us
Hello, sir. Sir, you are not audible.
I think there is a poor network.
Yes.
So he has left. We are waiting to rejoin. He will rejoin again. Sir is joining in a while. Please keep patience. Rupita ma'am, sir has joined.
Yeah, there is some power issue from my side. Just give me a minute, sir. I will.
Sure, sir.
Am I audible?
Yes, sir, you are audible.
Yeah, problem is bad. So, where were we in this slide when I got disconnected? Hello?
Yes, sir. Sir, this need for 3D bioprinting only, you were. The slide before, third slide, sir.
So, yeah. So, we need some models where we can evaluate the performance of drugs, okay, instead of animal models or some normal tissue cultures, okay, which are 2D cultures, okay. The problem is nowadays the ethical committees are also very sensitive they don't let us work on animal models directly because these technical advancements offer as a better platform, okay, to evaluate the effects of the drugs, or if you want to study a pathological condition, instead of animal models, we can have bioprinted equivalents. Okay, so the 3D interaction of the extracellular matrix with the cells is different in the bioprinted compared to the 2D cell cultures, which are made in the 2D environments. Okay. So these are all the problems in 2D cell cultures, as I told you earlier. We cannot exactly mimic the real physiological events which is happening inside the tissue in the 2D cell cultures. Okay. So we have in vitro models coming up. Okay. So they can be less expensive, less time consuming than animal models. Okay. Such models are under development to provide a mean of systematic, repetitive, and quantitative investigation of cell or tissue physiology in pharmaceutics and cosmetic industries. For example, you have made a drug. You want to evaluate the performance of the drug. We cannot directly give it to, as I mentioned earlier, we cannot directly give it to the clinical practice. Okay, we have to evaluate the drug in the in vitro models. Okay, so the in vitro 3D models, 3D printing gives us a upper hand. Okay, we are able to mimic the native human tissue, and that helps us in understanding the tissue morphogenesis and screening of new therapeutic techniques. Okay, so the idea is to represent the real physiological environment, which is the living human. Okay, so the current in vitro model consisting of 1 or 2 cell types lack multi-scale architecture and tissue-tissue interfaces, which are important in creating a functional system. Okay, most of the 3D tissue models suffer from lack of nutrition. So nutrition is supplied by the blood vessels, nutrition and oxygen. So not only the tissue or organ which is printed or fabricated, it should also have the vasculature, which will help to distribute the nutrients and oxygen. Okay, so we should have in addition to the mechanical model, we should have the network of vascular system. So that will help us to supply oxygen and nutrients to the cell. So when this networks are absent, okay, you can study only on the surface. Only planar or 2D cells can be studied. Because when you stack cells on top of each other, the cells which is deep which is buried in between, it lacks access to cells which is available in the surface. So the diffusion is also only few microns. Okay, when it is more than few microns, the diffusion of nutrients and gas doesn't happen. So unless you have a network, vascular network, it is difficult to give all the nutrients and oxygen to all the cells which are made in the 3D printing. So whenever we 3D print an organ, we include the vascular networks also. Okay. So if you are not including that, you end up analyzing only on a 2D planar cells. Okay. So that is also done for different applications. Okay. So what is 3D bioprinting? So bioprinting enables us to reproduce the complex multi-scale architecture by precisely positioning various cell types, biomolecules, and biomaterials simultaneously at predefined locations. Okay, so we have different address for each entity in our body. Okay, heart should be here. So within heart, this structure should be here. Similarly, within an organ, we have predetermined location. Okay, here the extracellular matrix should be there. Here the cells should be there. Okay, and then here some biomolecules should be there. Okay, so we have to position them at these locations. So that is done by bioprinting. Once you made it, like I told you, you have to include the vasculature also. So once everything is done, it can be put into the maturation chamber, where it gets matured, and it by getting the physical stimuli, okay, physical or mechanical or electronic stimuli, it undergoes maturation. Okay, so there are different materials which are used as bioinks. Okay, we have natural material and synthetic materials also. Okay, which provides us the base for the extracellular matrix. Okay, and then we can also take the extracellular matrix from a living human. We can harvest some tissues and then remove the cells from the tissue and take only the extracellular matrix. So that is called decellularized ECM. So that will act as a bioink source, which can be used in 3D printer for making the organ. Okay, so you need you can directly use the synthetic or natural or artificial material for which can act as an ECM, or you can harvest tissues and then remove the cells and then use that as also an ECM that can be printed. Okay, so this is the entire flow of having what you end up doing in bioprinter. Okay, initially you need the which organ you want to print, you want to image it, and then you get the structure out of it, and then you make a design layer by layer what you want. Okay, this is the final output. That design is taken from the imaging techniques, and then you layer by layer how you want the design to look, you make the design, and then you select the material, whether you are going with natural polymer or synthetic polymer, or you are using a decellularized electrocellular matrix. Okay, what the scaffold is made up of, you select the materials, and then you select the cells. What kind of cells you are going to use? Are you going to use stem cells? Are you going to use different cells which are harvested from adults? Okay, and then you mix everything and put it into the bioprinting nozzle. It will print layer by layer. So the printed 3D structure is ready. You put it for maturation. Maturation means you put it in a reactor, where in the reactor you give some stimulus, mechanical stimulus, and then you wait for the tissue to mature. Okay, and then you do in vitro testing, and finally you implant it. Okay, so fabrication methods, many fabrication methods are used. We use lithography, we use 3D bioprinting. Okay, so let us focus on 3D bioprinting in this modules. We can have other fabrication techniques also. Okay, again in 3D bioprinting we have different extrusion based method. Okay, you use pressure to fill the material in a syringe, and you use pressure to push the material which will come out of the nozzle. So it is pressure based, pneumatic based, or extrusion based systems are used. Okay, laser based systems are used. And then what are the sources of the polymer? Like I told, synthetic polymers are used often, but synthetic polymers lack the binding site which is required for cell signaling pathways. So we use natural polymers also in most of the cases. Okay. So we use collagen, silk, gelatin, fibrin, glycosaminoglycans, and other polysaccharides, which can be used as the ECM matrix. Okay. So decellularized ECM is also can be used. Okay, and then there are other cell sources also. Okay, you can take primary isolation and in vitro culture of primary cell cultures are difficult. Okay, when you harvest a cell, which is not a stem cell, when you harvest a human cell, the lifespan of the cell in vitro condition is very less. So you cannot plan studies for spanning over a few hours. If you want to do experiments for days, then the adult cells are not suitable for it. You have to go to stem cells. The hepatocytes isolated from the human liver have a limited lifespan in vitro. After that, they die. It is very difficult to maintain them alive. So better you take a stem cell and then induce or induce the differentiation so that you get the appropriate cell type. Okay. So, for example, in vitro culture and proliferation of glomerular podocytes are difficult. Glomerular podocytes are there in the nephron of the kidney. Okay. So, it severely hampers the in vitro modeling and functioning of the glomerulus. Okay. So, we go for cell sources like stem cells. Okay. So, we don't always have access to embryonic stem cells. We have adult stem cells also. But nowadays, embryonic stem cells are stored. Okay, whenever a baby is born, this is one private agency which does that. It cryopreserves the embryonic stem cells, and in future, if the person needs access to the embryonic stem cells, this company gives it. Okay, so this kind of facilities are available nowadays. Okay, so if this is not there, embryonic stem cell is not there for a particular person, we can take adult stem cells, pluripotent stem cells, and we can induce pluripotence. Okay. So as I assume that most of the people are from bio background, I am not going into different cell types, stem cell types. Okay. So what is the ability of the cell to develop into a whole organism or to develop into a particular organ system or to develop into a particular tissue? That determines whether it is totipotent, pluripotent. Okay. And how you can induce pluripotent in adult somatic cells. So these things are possible when you work on the adult stem cells. Okay. And then as I told you, we have to include the vascular network also. If the vascular network is not there, so the tissue will not get sufficient nutrients and oxygen and it will die. Okay. So we have to include this in the design itself. And then we use the bioreactor for maturation. As I told you earlier, you have to give the mechanical stimulant. For example, if you are developing a blood vessel model, 3D model, printed from a bioprinter, so it might require pulsatile flow because blood vessel is subjected to pulsatile contractions and relaxation. Okay, the vasoconstriction and vasodilation. Okay, so it is, and the blood flow because of the pumping nature of the heart. So there is a pulsatile movement of the blood vessels. Okay, so we have to give this as a stimulus. Similarly, in renal tubule and glomerular capillary, you might need compartmentalized fluid flow. These environments you have to simulate in the bioreactor. Okay, so in the case of neuronal tissue, so you have to give electrical stimulus. In the case of an orthopedic application, you have to give mechanical force or shear stress. So these are all the maturation chambers. So this is mostly for blood vessels. So you have fluids flowing at particular flow rates, and in this you put the developed blood vessels. So it undergoes maturation over a period of time. Okay. So this example is for blood vessels. Okay, the maturation chamber, the bioreactor. Okay, and then for other types, you have to give other type of stimulation, maybe an electrical stimulation, or if you are making a cartilage, you have to give mechanical loading in a cyclic way. Okay, so yeah, so this is the bioreactor part, which helps in the maturization of the 3D printed cells. Okay. So we will look at the different techniques which are used. So this is another example where you are where you want to make the proximal convoluted tube of the nephron. So you want to make that model, okay, 3D printed model. So you use some sacrificial ink to print a convoluted tubule, and then you put it in a cast. And then you dissolve the sacrificial ink so that you have a mold where the mold for the proximal convoluted tubule. Then within the mold, you inject the cells, and then the cells attach to the mold and they form a blood proximal convoluted tubule-like structure. And then later you give the shear stresses so that it is conditioned to real life shear stresses. Okay, so this is the entire process. Okay, so you can using this technique, studies have been produced a variety of in vitro models that accurately replicate the physiological function of natural tissues, including liver, heart, muscles, vascularized tissue, and cancer models. Okay, so what we saw example in the previous slide was the 3D model of a renal proximal convoluted tubule. Okay, so by removing the 3D bioprinted sacrificial material in a casted ECM hydrogel, a perfusable convoluted microchannel built in matrix was successfully achieved. Then you seed it with the epithelial cell, proximal tubule epithelial cell, and implementation of the perfusion signal. The engineered model exhibited significantly enhanced epithelial morphology and functional properties. So now the model is ready. Model of the proximal convoluted tube is ready. You can give any drug to it and see how the model is responding to a particular drug. So, for example, they are giving a nephrotoxin, cyclosporin A. So, the epithelial barrier is disrupted in the dose-dependent manner. Okay, so we can observe it very nicely in this model. So, if you want to evaluate nephrotoxin, better to you avoid animal model and directly practicing in clinical conditions. You can do on these kind of 3D printed models. It comes with own limitations. Materials, we are not that much equipped. Okay, we cannot use many materials, and we cannot use many processes in the process because cells are also involved. The cells may die in the process. We cannot apply high pressure. We cannot apply high temperatures. So these are all the technical. Whatever we saw in the polymer printing technique, we cannot directly use in bioprinting. Okay, so we have to develop some materials and develop some process which will help the cells to remain alive. Okay, so these are all three different techniques: extrusion based, inkjet based, and laser assisted printing. Okay, they are so this is you have a syringe and the liquid has the cells and the extracellular matrix, and you apply pressure, pneumatic pressure, and the cells and the liquid comes out and it is deposited on the bed. Here in the inkjet method, you make small droplets of the liquid and the droplets are printed on the surface using a method which is similar to inkjet printing. And then we can use laser. Okay, laser again, it involves heat energy, but we have to use it appropriately so that it helps us to deposit the cells on the substrate. Okay, so as I think I'm running out of time, I will summarize the techniques. Okay, I will share you this links. You can see them in YouTube. I think I have these videos. Yeah, so this is the comparison of three techniques: inkjet based, extrusion based, and laser assisted bioprinting techniques. So they support different viscosities. Okay, so if you have higher viscosities, the bioink is of higher viscosity, you can go for laser assisted technique. Okay, if it is in between, you can use extrusion. If it is very low viscosity, you can use inkjet method. And then how you make the individual globules. Okay, that is the gelation process. Okay, and then preparation time, it's different for different materials, and the printing speed is also different for different technique. Cell viability, okay, and then how much of the cells are going to be alive, and then what is the cell density which can be supported in each technique, that is also given. So this is a comparison of different 3D bioprinting techniques along with the working principle. And yeah, I will show you the working of, yeah. This is a pneumatic based, hydraulic based bioprinting system. Yeah. Yeah, this one. So is the video audio is clear?
Sir, for audio, you have to share your sound of the
Acha.
presentation.
I have shared the screen, but how to share the
Sir, during the sharing of your screen, there is an option to share your sound also. When you are sharing at that time.
let me then do
No,
it
no,
again.
this is not, sir. You just reshare again.
Stop share and reshare.
Yes, sir. Yes, sir. Yes.
Share.
When you will click on the share button, at that time, in the right hand side, there is a share sound button. So you have to
Okay,
click.
got it.
Yes, sir.
To talk to you today about the 3D bioplotter by creating the highest quality.
Now it is audible?
Yes, sir. Yes, sir.
At Desktop Health, we are committed to creating the highest quality 3D printers and materials for medical device and dental solutions. I'm really excited to talk to you today about the 3D bioplotter by Desktop Health, which is a state-of-the-art 3D printer for manufacturing of medical devices and other biosolutions.
The 3D bioplotter has been on the market for over 2 decades. We have 3 machines available: the starter series, the developer series, and the manufacturing series. Parts of the machines are designed and produced in Switzerland.
The 3D bioplotter is a very unique solution because it has 5 different print heads that can swap in and out, including those for UV curing, for low temperature materials, and for high temperature materials that can even go up to as high as 500 degrees Celsius, which is the largest temperature range on the market. Additionally, the 3D bioplotter has a temperature-controlled print bed where you can bring your prints anywhere from -10 degrees Celsius up to 80 degrees Celsius as you're printing. Its axes have resolution down to 1 micron, meaning that these movements are very precise. Additionally, there's automatic calibration of the nozzles via a camera system and also a nozzle cleaning system integrated into the unit. The integrated camera can record layer by layer for verification of your prints at a later date. The software is very easy to use, enabling users to program multiple materials together, including low temperature materials, high temperature materials, and support materials, all in a single part. The resolution of your parts is really determined by the nozzles that you're using, and we've successfully used nozzles as small as 100 microns in inner diameter for the manufacturing of the phonograph device, which is a novel biodegradable device for eardrum repair.
The bioplotter is a very intuitive machine to use. When guidance is needed, there are support teams to provide insight, which is important for tissue engineering applications when everyone's use of the machine is different.
The 3D printing process with the 3D bioplotter is straightforward. First, you upload a CAD file of your part into the software, and the software will then slice this into a print path. Then you decide what materials that your part is going to be made of. You can load a wide variety of materials, everything from viscoelastic liquids, hydrogels, photocross-linkable materials, and thermoplastics as well. Once these materials are loaded into the barrel and loaded onto the printer, then the printer can begin to print. It will start by taking the first material print head and calibrating the nozzle so that it knows exactly where it is with respect to the build plate. These materials can then be patterned layer by layer on top of each other, switching materials back and forth as the part requires. In this way, you can easily produce a complex part comprised of many different materials in a single print. There are 2 different support materials available for use with the 3D bioplotter. One for use with low temperature hydrogel inks and another for use with high temperature thermoplastic inks. Our team really likes that both of these ink systems are fully dissolvable in water. So what this means
This is how they use some solvents to remove the support material. So it is put in the liquid solvent and then it removes the support materials.
is that you can create a complex medical device with many different parts and overhangs on it, and then simply wash away the support material, leaving your part behind. At many Tier 1 research institutions across the globe, the 3D bioplotter is being used to move the nozzle in healthcare. There are researchers all over the globe developing custom blends of metals, ceramics, hydrogels, and unique print paths to develop complicated and reproducible medical devices that are going to go into a patient and improve their life.
Any doubts? I've come to the end of the chat box facilities. Any doubts, students? Sorry, participants?
The chat box, there is no question. But the participant, if you have any doubt, you can raise your hand or put
And
your
put in the chat box also.
in the chat box. Yes.
What is the participant profile like? More are from what background?
They are from bio background, sir.
Achha.
Sir, it was a very wonderful talk. I actually have a question. So I just want to know that can we incorporate some ceramics in this FDM, like the PLA or the thermoplastics? Can we incorporate some ceramics in these FDM wires? And can we print it? And like in what capacity, how much amount we can add the ceramics? Because actually one of the problem I have faced is that dispersing the ceramics in these polymers like PLA or PCL or is a very difficult task. So do you have any idea on this? Like, have you tried this?
Yeah, like if you are working from solvent and liquid state, it is difficult.
Yes.
So we have an extrusion system here. So
Okay.
in that it is more easy because we start in the bead forms and then we extrude it. So at that phase, if you add another one like ceramic or even metal, so it will sit in the polymer matrix and we can extrude a filament out of it, and that can be improved. So I also faced a challenge when I was trying to mix them at solvent level. I was trying to mix graphene oxide with PLA to get a PLA with graphene oxide, but I was not able to. But in extrusion level, yeah, I was able to. So sometimes I make them stick to the surface of the beads. So we have PLA beads. So, and I have graphene oxide powder. So I make them physically stick to the surface of the powder and then I extrude it with the extruder, which we have in the lab. And then the distribution is not uniform, but still I am able to get a composite filament. But like when I go to the solvent form, yeah, I'm not that expert in chemistry. So I tried in my lab, but we were not able to mix it. So that is also one challenge. Either you can physically mix it. And when you go to solvent-based technique, you are chemically mixing it.
Do you suggest any like some surface modification we can do, I think, and we can incorporate them so the blending will be more good, I think.
But again, yeah, it is a powder. So a powder you are adding to a bead. Both are physically adhering to each other. Sometimes the adhesion will come off. It will not be uniform. But it is possible. People have put metal powders in PLA and extruded out. People have put ceramics in PLA and extruded out. At the temperature in which PLA is getting printed, the metals and ceramics will not get affected. So they will be physically sitting they will improve the mechanical property or other applications will be there, but they will not react with the polymer because the temperature at which we are extruding the polymer, like ceramic and metal, will be unreactive.
Right.
It is possible, but in extrusion method, but again, the uniformity will be difficult.
Okay. Thank you so much, sir, for your excellent presentation today.
Thank you. So any other questions from the participants?
Let me check, sir.
You have a good crowd. It is more than 100.
Yes, sir. More than 200, sir.
200. Oh.
Yes, sir.
Okay. So I think participant can write in the chat box also. So we will move forward to our next station. But before that, on behalf of Department of Biotechnology and Bioengineering, School of Biosciences and Technology, Galgotias University, we thank you very much, sir. And I would like to ask Gaurav, sir, to present the e-memento.
Thank you, sir, for accepting our invitation. On behalf of our department, is Department of Biotechnology, Bioengineering and Biosciences and Technology, Galgotia University, as well as University of Cyberjaya, Malaysia. I'm glad to offer you this digital memento. Kindly accept as a token of gratitude. Yes.
Sir, I have a question.
Yes, please wait one second. So let me finish. So as a mark of our heartfelt appreciation for the sharing your expertise and conducting a great session, very informative session, sir, on the 3D printing and several aspects of the bioprinting. So we are highly grateful. We are honored to host you in this faculty development program. So kindly accept it, sir.
Thank you, sir. Thanks a lot, and thank you for giving me an opportunity to address this participants by this FDP. Soon I will ask Dr. Nishant also, and we will visit Galgotias in physical mode or any other event.
Yes, sir.
And interact with. Thank you, sir. So, Neha, you can ask your question. Neha, sir, no? Somebody was asking a question, no?
Yes. You please, you can unmute yourself.
I think she is not.
Participant, you can unmute yourself to ask the question.
Any other questions? She not responding.
Dr. Santosh Kumar ta bolte hobe. Santosh Kumar.
Rupita, you are muted.
So now it is now my privilege to introduce Dr. Santosh Kumar. Dr. Kumar is an associate professor and head of the Department of Chemistry at Harcourt Butler Technical University, HBTU, Kanpur. He earned his DPhil in chemistry from the University of Allahabad and has over 17 years of research experience, including prestigious postdoctoral fellowship in Portugal, South Korea, and China. His research focuses on biomaterials, polymeric functional materials, nanotechnology, nanocomposites, carbon dioxide capture and conversion, hydrogen storage, drug delivery, and environmental remediation. Dr. Kumar has established an outstanding international research profile with 118 journal publication, 19 book chapters, 5 books, and 3 international patents. He has successfully supervised doctoral and master's scholars led numerous internationally funded research projects, a recipient of the Research Excellence Award 2024 conferred by the Honorable Government of Uttar Pradesh, along with several prestigious international fellowships and innovation awards. Dr. Kumar continues to make significant contribution in sustainable materials, nanomedicine, and advanced functional biomaterials through interdisciplinary research and global collaboration. We are delighted to welcome.
Thank you, madam.
Yes.
Thank
So
you
we are
very much.
delighted to welcome Dr. Santosh Kumar.
Yes, sir.
Sir,
Thank
the
you.
virtual platform is yours. Thank you, sir.
Thank you very much. And first of all, I would like to thank Professor Gaurav Kumar, who has given this opportunity to present our research work in International Faculty Development Program. Today, my topic is Chitosan derivative emerging antimicrobial biomaterial for healthcare and sustainable application. So, slide is moving.
Yes, sir. Yes.
So, how we can get a chitin? So, we know chitin is the second most abundant biopolymer found in nature after cellulose, and it is also not soluble, non-soluble, but biodegradable. So its term chitin comes from Greek word is envelope. So where we can found chitin, you can see in chitin in nature, algae, fungi, protozoa, mollusca, annelida guts we can find from arthropoda, in cuticle, peritrophic membrane. We can find bacteria also. So in crustacean cell also we can find a chitin. So location of chitin, if you see in insect in Artemisia cyst, so here you can see if you cut section, so epicuticle, exocuticle, and endocuticle epidermis. So here you can see in protein matrix, chitin fibrillin is 2 angstrom diameter. And in Artemisia cyst, chitin containing envelope. So that's why it's called chitin in Greek word envelope. So a structure of chitin, if you see in carbon 2 position NaCO3 group, and this is the 3D structure of chitin. So where we can get chitin and chitosan. So how to prepare a chitosan. So if you take a crab cell or shrimp cell, and after decalcification and proteination with the acid treatment, we can get a chitin. And after getting chitin, you can use sodium hydroxide or enzyme treatment for deacetylation of chitin to get a chitosan. So this is the structure of chitosan. Here you can see in carbon 2 position in NH2 group. This is a very versatile group in chitosan. So this is the 3D model of chitosan. So if you see similarity and dissimilarity in cellulose, chitin, and chitosan. So here you can see cellulose. Cellulose in carbon 2 position OH group, and in a chitin in carbon 2 position NaCO3 group, and in chitosan NH2 group. So this is the dissimilarity. If you see in a carbon 6 position OH group in cellulose, CH2OH group here in a chitosan. So this is very similar in cellulose, chitin, and chitosan. And beta 1,4 linkage is also similar in cellulose, chitin, and chitosan. So if you see the property of chitosan, so there are many physicochemical properties like cationic polyamine. On protonation, adhere to negatively charged surface, mucoadhesive, and form gel with polyanion. And this material also you can use in 3D bioprinter, and it can form salt with organic and inorganic acid. It is also compatible and miscible with other polymer like cellulose. It has a high molecular weight linear polyelectrolyte, so it shows chelate forming ability with certain transitional metal, and it is also amiable to chemical modification because chitosan have reactive amino and hydroxyl group, and it is easily formed into fiber by electrospinning, and it has a high charge density at pH 6.5. So biological property, it has also biocompatible, biodegradable, safe and non-toxic. It has a bind to mammalian and microbial cell aggressively, and it has also shows fungi-static, spermicidal, anti-tumor, accelerate bone formation, and it has also hemocompatible, antibacterial and antifungal, and it has also used as a regenerative effect on connective gum tissues. So here you can see application of chitosan. So chitosan have a versatile application in food. So you can use as a food preservation, food packaging, and water purification also we can use. And some USA and some other European country, chitosan they can use as a fat binder. In material scientists use as a biosensor, bioimaging, and packaging film, textile finishing, polymeric membrane. In medical sciences, they used as a hemostasis, control drug release, wound healing, and wound dressing, anti-tumor, anti-ulcer. So in microbiological study and microbiological scientists can use as antibacterial, antifungal, and immunological also they can use as a biological response modifier and gene therapeutics, polymeric scaffold for cell culture. We have also worked on a gene delivery application of chitosan derivative, and there are many applications in agriculture to soil enrichment and to increase crop production, and this material also can use as a nano fertilizer. So nano fertilizer also can use, and flocculating agent, polymeric nanoparticle, synthetic polymer blend, and we can use as a chitosan. So here chitosan based biomaterial. So you can prepare with scaffold, hydrogel, membrane, nanoparticle. That material, scaffold, hydrogel, membrane can use as a bone regeneration, cartilage regeneration, dental regeneration, skin regeneration, and cardiac regeneration, and nerve regeneration. And also a chitosan material can use as a food packaging application. One of my student, she recently got PhD. She worked on a biopolymer matrix nanoparticle use in food packaging application. She used in tomato, chili, and grapes and ladyfinger. And it was also enhanced properties for shelf life of this product. So chitosan material can also be modified with chemical method by EDC NHS with graphene oxide. So we can also modify chitosan graphene oxide, and also functional multi-wall carbon nanotube. This material can use very application in gene delivery or carbon dioxide capture conversion and sustainability also. And the chitosan material we can also modified with functional multi-wall CNT and arginine, and also graphene oxide and arginine. So antimicrobial bionanocomposite. So many one knows about antimicrobial bionanocomposite. So this chitosan can use in a starch, with a starch, cellulose, and PLA, PVA, metal oxide, nanocarbon, nanosilica, and polyphenol. So this material can use as a food packaging application and some other wound healing, wound dressing applications also. And there are many techniques used in preparation of bionanocomposite, so solution casting method, coating method, also melt blending, and photografting method, electrospinning also. And antimicrobial mechanism, because we know chitosan antimicrobial activity has depend on many factors affecting like molecular weight, degree of deacetylation, degree of substitution of chitosan and other material, pH of the environment, type of microorganism like gram positive, gram negative, or fungi, and also concentration of the chitosan derivative and chitosan nanocomposite material. So there are so different derivative improve antimicrobial activity in different way. Like if you prepare a quaternized chitosan, so this quaternized chitosan has a permanent positively charged to provide a strong antibacterial activity even at a neutral pH. So and carboxymethyl chitosan also improved water solubility and better interaction with microbial cell. And sieve-based chitosan also we have also worked on sieve-based chitosan. So that material also enhance antimicrobial activity through a imine functional group and nanoparticle-based chitosan derivative also can enhance the antimicrobial property because higher surface area increase contact with microorganism and improve antimicrobial efficiency. So so here you can see chitosan material electrostatic interaction between chitosan and to negatively charged microbial surface, and also disruption of cell membrane integrity and leakage of intercellular content and penetration into cell wall, leading to inhibition DNA, RNA, and protein synthesis, chelation of essential metal ion, also disrupt the enzyme activity, and inhibition of biofilm formation, also in induction of oxidative stress, and microbial growth inhibition, and eventual cell death, also cell death. Here you can see collapse of the outer membrane of cell. So this combined mechanism make chitosan derivative and chitosan biocomposite material effective against a broad spectrum of gram positive and gram negative bacteria and fungi. And this so this type of material can use for biomedical, pharmaceutical, biomedical, food packaging, and wound healing applications. So we use titanium dioxide and silver titanium dioxide. So we synthesized in our lab. You can also synthesize titanium dioxide. It's a very simple method to synthesize TiO2. Sol gel method, so we can get TiO2 nanoparticle, and this is the silver loaded TiO2 nanoparticle. Can synthesize by co-precipitation method, and can also modify the chitosan gelatin TiO2 and the chitosan gelatin silver loaded TiO2 bionanocomposite film by solution casting. this film can use as a food packaging and wound dressing, etc. So this is also silver chitosan gelatin silver TiO2 bionanocomposite film. So this is the XRD of material. We confirm the structure of bionanocomposite material and modification of that TiO2 and silver TiO2. And this is the EDS analysis of TiO2 and silver TiO2. Here silver TiO2 is loaded 1.46 weight percentage. And this is the elemental mapping of chitosan gelatin TiO2. And this is the TM image of TiO2. Here you can see in a spherical shape. So spherical shape of TiO2 nanoparticle did not change after loading silver nanoparticle. Here this you can see dark portion we are located on the surface of TiO2 nanoparticle is the silver. So and this is the FESM analysis of bionanocomposite. This also, if you see this S1 is the chitosan. So chitosan is a smooth, homogeneous, and a compact surface and dense structure with very few pores. If you see here, pore and crack. And uniform polymer matrix due to a strong intermolecular hydrogen bonding. And this one is the starch S5 chitosan gelatin TiO2, and this is the chitosan gelatin silver TiO2. So this is the thermogravimetric analysis. This TGA analysis shows material is high thermal properties. And color parameter of bionanocomposite film we can use as and also see negative A plus A star is this one is the it means a composite film rate from 1.6 to minus 2.4 indicating a subtle greenish hue inherent to chitosan and gelatin. And this is the water uptake capacity and moisture content analysis. This is also shows material is useful in food packaging and wound dressing. And this is the antibacterial analysis of chitosan gelatin silver loaded TiO2. Here you can see this Staphylococcus aureus is a 12.33 diameter of inhibition zone, and E. coli 13.83 mm, and this is the diameter of the inhibition zone of E. coli. And this is the application food packaging application on green grape. So here if you see in after 7 days is also good condition grapes. And material in lab we have also modified chitosan starch TiO2 by nanocomposite film for food packaging application. So FTIR, and this is the FTIR of starch chitosan starch and pure chitosan and chitosan starch TiO2. And if you see here FESM analysis is also, this is the starch. Starch is showing granular or heterogeneous morphology, so more brittle than chitosan. And after modification with chitosan starch, here you can see it's this is a compact and continuous structures. And also it's a reduced crack and granule pore due to compatibility between chitosan and starch. And here you can see TiO2, it's showing a spherical and rod-like structure. And this in term image, if you see TiO2 is a spherical shape, and diameter is 15.53 nanometer. And this is the thermal analysis. This thermal analysis also shows a high thermal property of material, Chitosan starch TiO2, and DSC is also showing endothermic endothermic at 136.7 degrees centigrade. This is the porosity study, water uptake study, and water solubility, and this the moisture content. This also shows it's very good for food packaging. And this is the antioxidant antimicrobial analysis of a modified material. And then we also done with soil burial analysis of Chitosan starch, and Chitosan starch TiO2 composite material, and also food packaging application on chili. So if you see texture of sample Chitosan starch TiO2 dipped chili on day after 40, also texture of is very good condition. And we also you can also modify the Chitosan material with different type of derivative. So here we have also modified Chitosan bromine derivative gel. This material also very useful in antimicrobial activity. And you can see this SEM image. It's looking porous chain-like shape and cross section randomly oriented in a grain. And this material also shows more amorphous material. So that material can use in biomedical application. So we have also and also some derivative like Chitosan terephthaldehyde, Chitosan protonaldehyde. This material is also very good antimicrobial properties. So, and this Chitosan containing azo-based sheath base also shows very good thermal, antibacterial, and by reference properties for bio-optical devices. Also, this type of material can use. And here you can see inhibitory effect of Chitosan as control and derivative material against the E. coli and Streptococcus aureus. So this type also can modify it at room temperature. Diisocyanate modified Chitosans. This material also very useful in biomedical application, and it also enhance the antimicrobial property due to here you can see nitrogen atom. And here is the zone of inhibition. You can see 22 mm, and in Staphylococcus pyogenes have 20 mm diameter. So this we have also and you can also modify this Chitosan derivative with 2-acrylamide, 2-methyl propane, 1-sulfonic acid. This material make as a hydrogel, and hydrogel after solvent exchange you can get CS derivative gel or aerogel. And this material is also very useful in gene delivery application. And so conclusion is main synthesis of Chitosan derivative, aerogel, and bionanocomposite film, and its structure confirmed by FTIR, TGA, DSC, and X-ray diffractometry analysis. And thermal study shows high thermal stability than Chitosan, and the morphological study showed the derivative is uniformly the mesoporous structure and enhance the antimicrobial biomaterial for healthcare and sustainable application due to the nitrogen atom mesoporous structure and interaction size. And also our finding are encouraging as the nitrogen derivative can be used as a potential tool for emerging antimicrobial biomaterial for healthcare and sustainable application. These are my groups, and thank you. So any question?
Thank you very much, sir, for delivering such an insightful and engaging lecture. Participants, if you have any question, please raise your hand or you can type in the chat box.
I think there is no question.
Sir, as a token of our heartfelt appreciation and gratitude for your valuable contribution, we would like to present a digital memento. So, Dr. Gaurav Kumar, sir, please kindly present the memento. Yes, sir.
Yes, ma'am. The respected sir, please accept this digital memento as a token of honor on the behalf of our department, school, and university. So kindly accept it, sir. Thank you, sir.
Thank you very much, Professor Gaurav. Thank you.
Thank you, sir.
Now we would like to move on to our next speaker. It is my privilege to introduce the final speaker of today's program, Dr. Anusha Aditya, Chief Executive Officer of Exonics Biopharmaceuticals Private Limited. Dr. Anusha Aditya is a distinguished translational scientist, entrepreneur, and innovator working at the forefront of novel therapeutic development and targeted drug delivery for neurological disorders. Her research spans small molecules, therapeutic peptides, antibody drug conjugates, nanotechnology-based drug delivery systems, and advanced therapeutic platforms for diseases including glioblastoma, cerebral ischemia, spinal cord injury, and other neurological disorders. She completed her PhD at CSIR IGIB, where she focused on functional nanoparticles for biomedical applications. She subsequently pursued postdoctoral research at two of the world-leading biomedical research institutes, the Memorial Sloan Kettering Cancer Center and the Columbia University Medical Center, USA. In addition to her academic achievements, Dr. Aditya has contributed significantly to the biotechnology startup ecosystem by developing peptide-based therapeutics for metastatic breast cancer and now leads Exonics Biopharmaceuticals. We are truly honored to have such an accomplished scientist and entrepreneur with us today. So please join me in extending a warm virtual welcome to Dr. Anusha Aditya. Ma'am, on behalf of Galgutias University and the organizing committee, we sincerely thank you for accepting our invitation. And now the virtual platform is yours.
Thank you, Dr. Rupita, for that wonderful introduction. That's really nice of you. I'm going to share my screen. Let me know if you can see my slides. Yeah, are you able to see my screen right now?
Yes, ma'am.
Okay. So yeah, I know I'm the last speaker of today, and it's a rainy day in Noida and in the NCR. So please bear with me. get hold of some tea or coffee, and hope you enjoy today's session that I'm sharing with you, and hope you have some interesting questions
Yes,
as
ma'am.
well.
Actually, there are 300 participants with you. So
Okay.
you yeah.
I see.
Yes,
That's a
yes.
that's a huge crowd. So I'm very happy about that. You're still staying with me till this late evening, even though it's raining and it's a little bit sleepy, I guess. So thanks for staying with me, and I hope you enjoy my talk. So without much ado, I'll just proceed. I'll talk about two main projects that I've been working on for the past 5 years in Sloan Kettering, and then after that in Columbia. So in Sloan Kettering, I had worked on this uh, disease called high-grade glioma, which is very aggressive form of brain cancer. And we designed a silica-based nanoparticle which can cross the blood-brain barrier. And I'll take you through the process, how we designed this and what are the challenges we faced, etc. And based on that project, I also got selected in Columbia to extend that kind of project itself, but for other disease indications which involve crossing the blood-brain barrier and targeting other rare diseases. So let me take you through each of those projects, and hopefully you also find some use out of these. So the first project where I was working with high-grade glioma is about glioblastoma multiforme. It's a very aggressive form of tumor. And in most of the cases, it has been seen that only 7% of the patients, it has been reported, they can survive beyond 5 years. So it's a very aggressive form. And the major challenges that the biotechnologists and people in the pharma industry have faced while designing any therapeutic for this kind of cancer is a formidable barrier called the blood-brain barrier. Also, it's a complex network of epithelial cells, tight junction proteins, etc., which make it extra difficult to make any molecule pass this barrier. It is evolutionary prepared that way in our body, so that if we encounter any foreign pathogen or some kind of virus, our blood-brain barrier acts as the first line of defense. But that also becomes a challenge when designing a therapeutic, because more than 99% of the therapeutics are actually effluxed out of these barriers. And on top of that, when you're targeting a cancerous tissue in the brain, there's also a blood tumor barrier, which comprises of several leaky fenestrations, blood vessels, etc., which makes it even more difficult for any therapeutic to reach the target organ or the target tissue. Till now, the gold standard therapy is basically organic nanocarriers like liposome and a chemotherapeutic drug called doxorubicin. But the problem with this is the gold standard, but still the problem that people have encountered with this is very low bioavailability of this. There's a huge batch to batch variation. This also like the stability is compromised in certain cases. So these are all very big challenges for designing. So in my work, what we so before I started in the work in Memorial Sloan Kettering, there was already a lot of excitement going on around ultra small silica nanoparticles. So these are nanoparticles which are extremely small, less than 6 nanometers in size, and they have a very spontaneous propensity of crossing the blood-brain barrier because of its very small size. And on top of that, if you're able to conjugate on its surface with some certain peptides like RGD, or give protection through PEG, polyethylene glycol. So these multiple layers of protection can make these nanoparticles really superior and make them an ideal candidate for percolating the blood-brain barrier. So there was already some kind of work going on that, and these applications were used in melanoma mice model and in mini swine model. So we could see they were being used as imaging devices. These small nanoparticles, they were attached with fluorescent dyes like Cy5, like that, and you could actually see its localization in sentinel lymph nodes, like that. So this was a technology already going on there. So when I joined the group, the idea, we had a very dynamic group of biologists, clinical people, immunologists, etc., my postdoc guide. So the idea was to use these same kind of imaging devices as a drug delivery agent for doxorubicin, and at the same time we wanted to develop this in a way that these silicon nanoparticles can also make an immunomodulation of the cancer microenvironment. Now, why this is important? Because these tumors are also it's known as immunologically cold, because it doesn't have access to a lot of immune cells reaching those tissues, and so hence the immune system is not able to counteract the cancer tissues by itself. So our project was also to design these nanoparticles in a way that they supercharge this microenvironment, and now they are converted from a cold tumor to a hot tumor, and now the environment is more conducive towards accepting these immunotherapies or chemotherapies. So it makes a very nice combination therapy, and makes the a more efficacious route of working. So how this was created, the small C dots or the silicon nanoparticles, on its surface we used a kind of chemistry called azide chemistry. It's a covalent linkage through which these doxorubicin molecules were attached on the surface of the C dots, as well as targeting peptides like RGDs. It was very clearly seen through just colorimetric view that the ones which were unconjugated were cyanide, like kind of bluish in color, but when they were conjugated with the doxorubicin, because of their own color, this turned into a brownish kind of fluid, and from this we could kind of make a calculation of drug particles per nanoparticle. So that kind of calculations were also done. We had an idea that there were around 35 molecules of doxorubicin per particle of these C dots. So the next logical step was taking it in vitro cultures. So we were checking in RCAS cells, which is a cell line model for high-grade glioma, and we checked that in the regular cells, when in the RCAS cells, when we are just administering the unconjugated nanoparticles, we don't see a lot of fluorescence or uptake within the cells. But when we are administering only like free doxorubicin, there is some uptake. Now I'll make a comparison with the doxorubicin conjugated nanoparticles, how that is better than the free doxorubicin. So basically the doxorubicin nanoparticles, as you can see, there's a lot of localization across the whole cell as well. We're actually localizing in all the parts of the cells, and it was also not damaging the cells in a way that they are ruptured completely. There was a targeted delivery, and with time it was releasing the doxorubicin, and then the rupture was happening. But the problem with the free doxorubicin is that it was just entering, and within 20 minutes there was rupture of the cells. This was a concern, that because of this kind of mechanism, probably the regular cells, the normal cells, might also get affected. So that was a problem. So following that, we also did a cytotoxicity study on the RCAS, and we did IC50 calculations, and we found that our doxorubicin conjugated nanoparticles had much more higher efficacy compared to the regular doxorubicin. So this was a very encouraging result at the cellular level. Next, we took this study to mice level to make sure that we are able to actually cross these blood-brain barriers. So in a normal health, we did the study in both healthy mice and a tumor-induced mice. So in the regular mice, with the administration of the zirconium, zirconium is the radio label, zirconium labeled ultra small silica nanoparticles, we could see there was a biodistribution across various organs, and that is quite expected. There's always, whenever you're administering a nanoparticle intravenously, there is localization to liver, spleen, stomach, all the different organs. But if you see, there's a 0.04%. Now, by numbers, it seems like 0.04% of the injected dose reaching the brain is very low, but trust me, it is better than any other drugs where even the percolation is like in fractions. So it's usually doesn't percolate beyond 0.01% as well. So this was kind of very interesting to us. And then we also conducted the study in multiple GBM models, and in that we could see even higher accumulation of these nanoparticles in the brain tumor area, and this was actually retaining in the brain to at least till 72 hours. So that was enough for the nanoparticles to deliver the doxorubicin and perform its functions. We also did a histopathological study where we sacrificed the mice and took out the brains and saw a lot of localization only in the tumor area compared to the non-tumor areas in the brain. So this was also confirmed through MRI scans that this is the area where the tumor is present and our nanoparticles are specially accumulating in the brain tumor area. So this was biodistribution wise, we were very happy with that, and we took the study to the efficacy level. So here we did a survival study on the mice. So first we did a histopathological study where again we checked that okay, our modified nanoparticles are reaching the tumor level, and then we checked through a survival study that actually this is the conjugated nanoparticles are increasing the survival of the mice by at least twice of the original. For example, the only glioblastoma mice were surviving for only 15 days post induction of the tumor, but the modified nanoparticles could show the mice staying alive till beyond 25 days. So this was a significant jump in the survival efficacy. Following that, we also checked the tumor burden in a histopathological study, and we could see that there was complete elimination of the tumor compared to the ones in which only doxorubicin was administered. So this was confirmed through HNE, and Ki67 is a proliferation marker. Through all those markers, we confirmed that our nanoparticle was able to deliver, pass through the blood-brain barrier as well as eliminate the tumor and increase the survival efficiency in the mice. So this was very encouraging for us. We wanted to also want to understand what is the mechanism through which these nanoparticles are acting. So one of the idea was maybe these nanoparticles are targeting a pathway called the gamma H2X pathway, which is basically targeting the DNA damage, basically inducing the DNA damage. So gamma H2X is a marker of the single strand DNA damage of the cells. So we could see that there was high expression of the gamma H2X with the induction of the modified nanoparticles, and this was significantly higher than the vehicle or the only doxorubicin treated ones. And as I mentioned before, our premise of the study was also to make sure these nanoparticles not only cause the elimination of the tumor, they also supercharge the tumor microenvironment in a way that now it is more conducive to other forms of treatment, for example, immunotherapy. So for that, we made sure we checked two different markers. One is IBA1 and CD206. So IBA1 is a marker for microglia, and CD206 is a marker for polarization, macrophage polarization. So we could definitely see that with the doxorubicin, there was more polarization towards the M2 macrophages, but with the dox modified ones, there was less polarization of the macrophages. So this was an indication that basically it is causing a kind of supercharging a little bit the environment, but also not damaging the or causing any special side effects at the same time. So to make sure that our therapy is also safe and efficacious. So this was the study. We also checked that there are less side effects. One of the major problems with doxorubicin is that it induces damages to the cardiac tissues. So we also, after the sacrifice of the mice, after the whole treatment regime, we isolated the heart of the mice, and we checked that with doxorubicin there's a huge induction of the gamma H2X, so which is a marker that there's a lot of stress which is built. Stress molecules are released due to the doxorubicin administration. But once we are administering dox NDC, the modified nanoparticles, that kind of stress levels are dropped. So which means that the side effects which are usually caused by doxorubicin is much less in comparison to the one that we are administering. Following that, another major marker of the side effect and the effect in the heart is the troponin level. So troponin levels are also correlated to cardiac arrests in many cases. So right after the administration, we wanted to make sure there's no chronic, there's no acute or chronic effect because of the nanoparticles. So we could see actually with the administration of both the type of the free doxorubicin and doxy-dot, there was some induction, but in case of doxorubicin, this amount of production of the troponin levels were much higher in comparison to the doxy-dot. So this is, to the left, you will see this is a calibration. We wanted to see at what level the doxorubicin release is the highest. So once we captured that, we saw that the highest production after dox administration of the troponin levels is within 1 day, and then it drops. So we captured the next ELISA assay in the first day after administration, and we could clearly see a difference between the troponin levels in these two. So from all these studies, it indicated that our nanoparticles were efficacious. At the same time, they were showing less side effects in terms of high troponin levels or gamma H2X levels in the heart. So yeah, so further this study was very was taken further by the following postdocs and all. They wanted to see what exactly has happened. We alluded that okay, it's mostly going through the gamma H2X pathway, but there was another pathway involved called the ferroptosis, which we did a preliminary study and saw that there was a high expression of a gene called LAMP1 GFP with the administration of these nanoparticles. And this was a marker that, okay, that maybe it's going through the ferroptosis pathway. So the following studies were to link whether this supercharging of the tumor microenvironment is linked to also the ferroptosis pathways and how it works, and whether the slight injury that's been caused is also helping the tumor microenvironment to go towards a more hot microenvironment, immunologically hot microenvironment. So that study is still ongoing, and hopefully we'll have those papers out also soon. So this was an auxiliary study. Seeing these results of our nanoparticles, we got a lot of traction from different industries, and AstraZeneca, one of the major pharma companies, they were also very interested in this technology. So they sent us a linker basically, and we had a single chain fragment, HER2 fragment basically, which can be used towards targeting gastric cancer. This technology can also be targeted towards breast cancer, because most of the breast cancers are HER2 positive. But at that point of time, our project in hand was on gastric cancer, so we went forward with that. So I'll just briefly touch upon this. We saw like a higher concentration of the HER2 conjugated and the linker loaded nanoparticles in the HER2 positive cells. And compared to that, when we checked also the HER2 negative cells, which is a triple negative breast cancer cell line, which has all the all the markers like HER2 and two other markers also missing in that, we could see less localization on those type of cells. So this was also very exciting for us. We could establish that our nanoparticles were specifically targeting HER2 positive therapies. And then efficacy, why we did specificity studies, and after that again biodistribution studies and efficacy studies. Biodistribution wise also, this was a xenograft mice model where this particular gastric cancer cell line was injected subcutaneously, and we could see our nanoparticles through PET distribution was mostly localizing to the gastric tumor tissue and not to other organs of the body. So this was very encouraging. We did like tumor elimination studies completely. This is subcutaneous, so you can see the control fragments. In those mice, the tumor is still remaining even after 7 days of administration. But the ones which were targeted with the particular HER2 ones, the tumor was completely removed and it was eliminated from the system. And there was a longer study where the relapse was also checked over a period of time, over a period of 2 months, 3 months. It was also seen that there was no relapse of this tissue in case of the targeted nanoparticles. So this was again very, very, the pharma industries were really happy with these results. So this resulted in some publications, of course, and with the industry as well as with, a spin-off from my supervisor's lab. So that was the taking from here. This was a group. We went to a AACR conference after that. So our work was very well received. So this was the first part. And then I'll just briefly, I don't know how much more time I have, probably like I have 15 minutes more, I guess.
Yes, ma'am. Yes, ma'am. You can take.
Yes, ma'am, continue.
Okay, sure. So yeah, so this was all the work that was going on Sloan Kettering Institute, where we were working on cancer mostly, gastric cancer, glioblastoma, prostate cancer, all different indications. But then because these technologies were helping to cross these formidable barriers, especially the most difficult barrier, blood-brain barrier, this project caught the attention of a team in Columbia also, Columbia University. So they were working on a rare disease. So this rare disease is called globoid cell leukodystrophy. It's a little difficult name, so don't get scared by that. I'll take you through that. So this is a rare disease, and they were very interested to find some kind of technology that can help them. They already designed protein-based drugs, and they already designed some small molecule-based drugs, but they couldn't, in the animal models and in patient model, in all these clinical models, they were not able to achieve the crossing the barriers. So they sought some help from our group and me. So I also started working here after that. So let me give you a brief idea so that you're clear about what is the disease and what is the work. So this is an inborn organelle disease which affects the white matter of the brain, and it results in severe disabilities or like you can, as people mention them as special kids. So this kind of white matter diseases affect them. The prevalence is low and it depends on demographic areas. It's an autosomal recessive disorder, and it is characterized by neurodegeneration, severe walking disabilities, and even like premature death. So this whole umbrella of diseases is known as lysosomal storage disorders. This happens mostly because there are some metabolic changes which happen, and there's some toxic metabolic products which build upon in the brain and results in these kind of neurological manifestations. So, and this very briefly, the biochemistry behind these LSDs. It's also known as sphingolipidosis because there's this accumulation of this certain sulfatides, and there are this glucocerebrosidase, etc., ceramides, etc. These are sphingolipids, and there's huge accumulation due to absence of certain enzymes, and these lead to diseases like Krabbe disease, Niemann-Pick disease, Fabry, Gaucher, Tay-Sachs, etc., which all come under the lysosomal storage disorder umbrella. My work was mostly focused on this particular enzyme called the galactocerebrosidase, and it resulted in this disease called Krabbe, or the globoid cell leukodystrophy, or GLD. So basically what happens, this particular sphingolipid accumulates a lot due to the so there's a particular metabolite called the galactosyl sphingosine or psychosin, which accumulates a lot due to the absence of this particular enzyme called the galactocerebrosidase, and that leads to this neurological disorder. So in terms of neurological manifestations, there are initially in the kids, there are very simple symptoms like just crying and irritability, fisted hands, poor head control, like that in the early stages. But as it progresses, people start seeing seizures, loss of vision, hearing and voluntary, lack of voluntary movement, things like that. And with the late infantile ages, there's much more progression. In some cases, these manifestations come very later at life, even like beyond 16 to 30 years of age. Suddenly it starts showing up where there's some vision problems, muscle weakness, etc., and suddenly it progresses and to cases like seizures, epilepsy, cerebral palsy, spasticity, different kind of manifestations show up. In terms of like the pathogenesis, as I mentioned in the earlier slide, there's high accumulation of this particular enzyme called this particular metabolite called cytosin, and that shows up in the macrophages, where it takes a structure of globoids. That's why the name comes like globoid cell leukodystrophy. So because of the shape of the macrophages that take place under the microscope, because of the accumulation of these toxic metabolites. So going on to the pathophysiology, because it affects mostly the central nervous system and also peripheral nervous system in cases. So the primary targets which are seen, like what is going on in the brain, why is this kind of physiology showing up? And it's seen that it's mostly because this particular kind of cell called oligodendrocytes are affected in this case. There's a multiple population of cells in this in the brain, of course, like neurons, microglia, astrocytes, etc. But the one that contributes the most to the formation of myelin sheaths is called the oligodendrocytes. But in this case, what happens, these oligodendrocytes are unable to produce enough amount of galactocerebrosidase because of a particular mutation, and because of that, the myelin sheath is not formed properly, which leads to all these white matter disease and subsequently all the neurological problems. As I mentioned earlier, it affects these two major organs, central nervous system and the PNS, which is the peripheral nervous system. So the manifestations happen in terms of like these globoid cells, as well as like neuropathic pain in the periphery or peripheral organs. So the study that we designed over there is that we have to replicate this whole phenomena that is happening on a plate, on a cell culture plate, and we have to do it, the whole the Krabbe disease, we have to replicate that, the oligodendrocytes which are affected in a cellular environment, and then do a high throughput small molecule screening, and then see whether these small molecules can be delivered to the brain. So that was the whole premise of the designing of this project. There are animal models present. The spontaneous mutations have occurred in mice and even canine models and primate models. It has been seen, so non-human primate models. But the problem again, like I think speaker just before me or before that, he mentioned like using all these animal models for work is being prohibited, restricted a lot by the FDA now. There's some more push towards building organ on chip kind of models, or trying to see if you're doing this high throughput screening type of studies, whether they can be conducted in cellular models. So those are the ideas which are gaining more traction these days. So we generated a cellular model of the neuronopathy that we are seeing in the patients. So what we did, we received the fibroblasts, so basically skin biopsy samples from the patients, from the hospitals. These patients were already having this Krabbe disease or GLD, and then converted the skin biopsy samples to the fibroblasts, and then through a producing a stem cell based platform through it and then converting it to oligodendrocytes. Now, classically, there were papers already published in this field where people have done this conversion in a two-step process. First, converted from fibroblasts to induced neuronal stem cells, and from these stem cell kind of culture, various kind of pathways can be taken. Either neurons can be generated, or glias can be generated, or oligodendrocytes. So there's a whole small molecule based protocol for producing these oligodendrocytes. But the published protocols were very time consuming. It's like 3 months or something to get from the fibroblast level to the oligodendrocyte level. And also, it was also very expensive process. So due to to make it more time efficient and cost efficient, we designed a process where we can do a direct reprogramming from the fibroblasts to the oligodendrocytes. So to do that, we did a combination of small molecules, small molecule induced differentiation, as well as we used a particular plasmid which was producing three important stem cell inducing factors. They were SOX10, OCT4, and NKX2. So these three were cloned in a plasmid and then added to the GLD fibroblasts. And for around 7 days post the induction, they were still in puromycin selection, and whatever culture was present beyond that was taken for oligodendrocyte differentiation through particular chemical differentiation factors. So just briefly how this is done is like, once you already have the oligodendrocyte reprogrammed cells, you can again pass it through this technology called MAX. Also, like it's quite equivalent to FACS, but it's magnetic activated cell sorting, and it uses a particular magnetic bead which has O4, which is a protein which is highly expressed on the surface of oligodendrocytes. So these magnetic beads, they capture specifically only the oligodendrocyte population. And then subsequent, the characterization of these cells were done through PCR and then cytochemistry and, plate-based, chamber slide based myelination assays. So that kind of work was conducted. And then we did the characterization. As you can see, the control fibroblast cells, they didn't carry these O4 positive, the fibroblast ones, but after conversion, there was a huge, expression of the O4, which is a classical marker for the oligodendrocytes. We also made sure other factors like oligo2 is also highly expressed. SOX10 is another marker we checked that is also present in higher quantity, CNP. So these are all just like characterization to see. Also, we made sure that these cells are not moving towards any particular, neuronal or glial form. So, nestin is a neuronal marker. So, in that case, we didn't see a lot of nestin expression. We were seeing more of O4 expression with the conversion. So, this was interesting for us. Following that, we, after doing the study in just one cell line, we progressed it to different, 7 different cell lines from 7 different patients, all clinical, samples, skin biopsy samples, and then they converted to fibroblasts. And we also checked what kind of mutation they had, whether it was early onset, whether it was late onset, and whether our technology is being we are able to replicate this technology across different patient cell lines. So this was also proved through our technique. And like I mentioned about the max before, we isolated the O4 negative positive cells, and we could see there was actually the conversion efficiency was quite high in case of all different, across all different populations, control or early onset or late onset. Especially in early onset, there was a huge conversion efficiency. So this was something that they really interested us, that this technology is like a very robust technology. We did a gene expression analysis. So basically, like I mentioned before, the reprogramming pattern. So it starts with the neuronal stem cells and oligodendrocyte precursors, but our ultimate goal is to see whether we are able to produce an oligodendrocyte which is myelinating. That will be our final goal. Just make sure that we are able to reproduce the complete biology on a plate. And we are trying to, we can do a phenotypic analysis also. Okay, these cells are actually myelinating. So we checked across different markers for the different stages, and the classical markers, oligo 2, OSP, CNP, they all seem to be very highly expressed in case of both the kind of type of fibroblasts. So, and also we did an this cytosine expression analysis. So this is basically the toxic metabolite, and we try to see if the accumulation is also being replicated similarly like in the clinical case. So we saw a very high accumulation in case of the early onset ones. The late onset one study is still going on. We didn't initially see a lot of cytosine expression, and that pathway we are still investigating why. Maybe in case we probably may not be able to see the late onset cytosine completely, but this part of the study is still ongoing actually. And then, as I mentioned, the myelination is the final goal that we want to see. So we could actually see, so this is a chamber slide with nanofibers actually. And on the surface of those, we plated our O4 positive oligodendrocytes. There was a high expression of the MBP markers, which is the myelin binding protein. So high expression of that marker makes sure that, okay, these cells are actually producing these kind of myelin over the nanofibers. So that was encouraging. And then we also did a 3D reconstruction to see which all places these myelin are like branching out, and whether, so the final goal was to study with the administration of small molecules whether they are able to break down these myelin formations, and what is the pathway basically behind that. So this brings me to the small molecule screening study. Just for a background, such kind of high throughput studies has been done for these, these kind of diseases, lysosomal storage before, but they were mostly done on fibroblasts. People were not able to achieve those in oligodendrocyte because obviously it's not so easy to extract brain tissue from a healthy kid or a kid, a diseased kid. So it's not a feasible method. So people conducted those studies either on mice tissues, or they have conducted those on skin tissues. So none of them were actually recapitulating the real biology, but it was good for starters. So these kind of study were conducted in a lab before, in around 2013 2015, that time. So now with the new technology, with the new properly characterized oligodendrocyte cells, we did study of the small molecules through a high throughput screen. Just to mention, in the previous fibroblast study or mice derived tissue study, there were like top 16 candidates which were identified, and those 16 candidates we basically produce analog hits through collaboration with our chemistry labs, and they helped us generate these hits for especially for our particular study. And in this study we found two main hits which were able to increase the enzyme level, the enzyme activity, which is the galactocerebrosidase. This was very encouraging, because either we were trying to target two pathways. Either it should increase they should help the decrease of the misfolding of the galactocerebrosidase enzyme, or they should help in proper refolding. So that was our hypothesis. The other thing that we were also trying to see, that what is the end result, whether these 16 analogs are also able to decrease the toxic the cytosine pattern. So this was two of the hits also showed us that they, through the dose response curves, that they are actually able to decrease the cytosine levels. So we were trying to come down to some molecules which can perform both the activities, maybe increase the enzyme activity and also decrease the toxic metabolite production. So this is the technique through how the small molecules were identified. The next idea was, like always, the biggest challenge is crossing the blood-brain barrier. So what we did is a special kind of nanoparticle was used in this case. This is called exosomes. So exosomes are cell-derived nanoparticles, basically. So they are produced by culturing the neuronal cells for a longer time, and then you take out the supernatant, basically, and pass it through the ultracentrifuges and size exclusion chromatography, and then you come down to a size range of about 50 to 200 nanometers. So this was characterized through transmission electron microscopy, and we found that this might be an ideal disk because it's also biocompatible. It's generated from the neuronal cells only. So these can be quite ideal candidates for either carrying the GALC protein, the galactocerebrosidase protein, or the small molecules that we have derived from our studies. So this was the next part. So this is the workflow of isolation of the exosomes. If somebody is interested, I can discuss more how the labeling part is done, and can share protocols on that. And there's also a lot of publicly available literature also. The main thing for characterizing these exosomes, that they have certain membrane markers like CD63, CD81, etc. So we wanted to make sure, these are also known as tetraspanin markers. So we wanted to make sure that the expression of all those markers are intact, and we are in fact only isolating the exosome population. It was also seen that spontaneously, when we are deriving it from these cells, spontaneously these exosomes are also carrying as a cargo inside them these galactocerebrosidase proteins. So this was very encouraging, because expression of these proteins organically helps us just to administer them directly, instead of again encapsulating some another biologic separately. So that was a win-win for us, kind of. So again, we needed to check this abundance. So we chose lymphocytes because they have quite a high uptake efficiency usually. So we wanted to see what is the distribution in these lymphocytes. We characterize these nanoparticles as around like 120 nanometers in range, like that. Also, like the characterization, as I mentioned before, through Western blotting was done. And then, yeah, we try to see what is the abundance across a time frame. So we saw like it was the highest peak uptake in the cells is around 4 to 6 hours, and then there's a slight drop till like 24-28 hours. There's a drop in the exosome abundance through with time, which is also expected because you want the nanoparticles to perform its function and then release itself automatically out of the system through liver and spleen, because you don't want them to accumulate in the circulation for very, very long until and unless you're very sure that it has no side effects at all. So that was a study. And next we went for the biodistribution studies as well. So the Twitcher mice is the classical mice model for the Krabbe disease or the global cell liquid GLD that I was talking about. So the Twitcher mice, they are named that way because they have an efficient manifestation of like constantly twitching their forefeet, and they do not survive also beyond 45 days because of this lack of this enzyme. But we managed to do a quick study on these mice, and we could see there was a high, there was a lot of like also like mice to mice variation, but in most of the mice we could see like there was accumulation of the nanoparticles in the brain. So we are still like refining this technology. We are seeing what is the way like we can achieve like a higher efficiency. Like in most of the mice this blood-brain barrier can be crossed, and it reaches it performs the activity properly. So yeah, again brain sections were taken out, and we were checking whether the fluorescently labeled exosomes exist in those sections, and there was also quite a lot of biodistribution across the brain section. So this was also something that we were expecting to happen. So yeah, this study also came to an end, but being followed up by other students, other postdocs. So they are also doing further small molecule screening. We're trying to see which mechanism of action is best for targeting this disease. And then we are trying to see whether this same technology can be also replicated for the peripheral nervous system diseases. So these studies are still ongoing. So while this was going on, all these studies on like trying to see whether we can treat certain rare diseases, whether we can cross the blood-brain barrier, target glioblastoma, I also tried to see. So this is like I need to acknowledge first the group in Colombia, but as I was mentioning that beyond that, the idea is always to see what is happening at the global range. So our technologies was getting a lot of traction from the pharma industries. Sanofi collaborated with us for the last project that I described, and Sanofi, they gave us a registry. They have a registry of all the patients across the world who are suffering from lysosomal storage disorders, and what is the prevalence, what is the disease abundance across different countries. And I saw, of course, like because being an Indian, I had all the eyes on what is going on in India, and I saw that there is also a lot of prevalence of these rare diseases, as well as lysosomal storage, white matter disease. There's a lot of prevalence here, but there's less awareness around rare diseases, and the early diagnosis, all these things are a challenge still happening here. So I was thinking in my mind what we can do about it, like whether we can approach a broader crowd here, try to make some awareness around these diseases, try to find some therapies around that. So all those were going on in the mind. I tried to make a map. I also, went through the literature to see, which all states are, having these different diseases, what is the distribution. I reached out to certain organizations, philanthropic organizations, and AIMS New Delhi was very, like, they were very helpful because they have a whole program on lysosomal storage disorders going on. So they were also excited about our project, and we had some collaboration built with them. So I thought, like, it's time, like, let's make a move and let's try to see what is going on here, and whether I can contribute some of my training and some of my knowledge to any of these crisis happening here. I saw a huge market. Like, I didn't find any market study done in India as such, but I saw a study from China. I found, like, this is also a huge, like, $1 billion market. So I was like, why not, let's try to see if we can build a startup around this, and try to also gain traction in the way, like, collaborating with pharma industries, and let's see if we can capitalize on this area. So that's where Exonics started. I founded this company in January this year. It's still very, very new. But I'll just briefly, like, just go through what's going on here. I mean, because of all this training in demyelinating disorders and white matter injury, I thought that there's a broad spectrum. My training has been on leukodystrophies, but we can also expand it in other indications like multiple sclerosis, neuromyelitis optica, and other demyelinating disorders. So I saw there's a huge market over there, so which we can try to address. The target biology, I already, it's pretty much similar. Oligodendrocytes are affected, and there's this problem of demyelination, which leads to certain disabilities. But if we are able to produce something which can help in remyelination, either in the form of small molecules, or in the form of protein biologics, or CRISPR-based, mRNA-based therapy, which can help this remyelination of the oligodendrocytes, then we have an edge, and we can support and help a lot of patients in that case. So, and I also checked what are the existing therapies. The existing therapies are mostly like targeting, because the multiple sclerosis, for example, is an autoimmune disorder. So they're just targeting the autoimmune environment and trying to develop monoclonal antibodies around it. But that only tackles the symptomatic problem at that point. But if there is relapses and the myelin sheath, which has already degraded, that part of the problem is never really addressed. So the person who has already reached a point of pain or disability, they can only do a symptomatic management, but reversal is never happening. So the idea was to target all those things, whether we can achieve a reversal of that situation. So people have used steroids, enzyme replacements, etc. But all the problems, like steroids have a lot of side effects, as we know. Enzyme replacement is, again, like, it's very expensive. So I also wanted to make this a cost-effective kind of therapy. So based on those ideas and the trainings, I understood that there are two critical bottlenecks for preparing that kind of drug, remyelinating drug. One, as we talked about, this formidable layer of epithelial cells and tight junction proteins in the form of blood-brain barrier and blood CSF, cerebrospinal fluid barriers. So, and it fluxes most of the small molecules or nanoparticles, whatever it is. It effluxes most of those things. And also, there's no patient-derived high-throughput screening platform. present. So these are two main pain points still for preparation of these kind of drugs. So because of one by one, I tried to address each of those pain points. The first, to solve the first problem, was designing of this conjugate called NV Trinity. I just mentioned named it after one of my favorite characters, because I really believe that this can help in rendering a lot of all these disease manifestations can be cured through this. Again, we are using exosome technology over here, either in form of exosome or other nanovesicles, but because their tendency of spontaneously crossing the blood-brain barrier, that is option A. And then we are also conjugating on the surface something called a nanobody, which is only the heavy chain of the of an antibody, and these are only present in some specialized organisms like camelids, etc. It's very new, and like there's a lot of excitement around this kind of nanobodies, because many studies have shown that they are more specific and more effective than regular antibodies, monoclonal antibodies. And because of their small size, they can be easily conjugated on the surface of the exosomes. So I collaborated with a group who are already working with these nanobodies, and designed this transferrin integrin expressing nanobodies, which can help in crossing the barriers. And also the idea of preparing cell-derived nanovesicles came from the fact that they can be scaled up very easily, because when you're thinking from a industry point of view, you also always have to think about scalability. You have to think like your whatever product you're developing must be it's very scalable and also has less batch to batch variation. So all those things, when you're in mostly an academic setup, you only think about like just to see what is the best way this can work. But when you're in an industry setup, you also have to think about these additional layers of complexity, like scalability and profitability, of course, so all those things. So this was addressing the first pain point. And the second pain point, of course, like we discussed before, that we want to derive a platform which can help in remyelination therapies. It's going to be similar to what we derived before in Colombia, but of course, we are getting the patient samples here in collaboration with AIIMS New Delhi. And also the technology is a little varied in this case, because here we are not only focusing on leukodystrophy, but we are also focusing on all kind of remyelinating disorders. So it's a little modified from the previous technology. So there's a huge market landscape. There's competition from US and UK, but there's not as far as I have screened, there's not a lot of companies in India working on blood-brain barrier and all these problems, neurological problems. So it's a niche area, and there's a lot of opportunity in this space. And of course, the most important thing is it has a lot of socio-economic impact, which is we want to take our technology to patient, caregivers, biotech companies. The ultimate altruistic goal is to help the patient, decrease the cost, make the drugs more cost effective, because right now the existing therapies for multiple sclerosis also is very, very expensive in India. And also job generation, help women to come to the workforce. So all these socio-economic aspects also we are very excited about, and we want to contribute on those grounds as well. I have a wonderful team. My PhD guide is also backing me up. We have Dr. Nija Gupta from AIMS, who is a pediatrician and has a lot of experience in lysosomal storage disorders in India. And Dr. Arjun Ray from IIIT, he's also helping me in we are designing some AI enabled remyelination drugs. So he's a computational biologist. He's helping me in all those areas. We have a business advisor, and there's backup from IIT Delhi. So we are very, very happy about our team. Luckily, this projects were also had a lot of attention from the Indian government, and they awarded me the Women's Leadership in STEM Entrepreneurship Award, and like 20 women across India were selected for this program. And we recently had our immersion program in Hyderabad, so they are helping a lot, like first generation entrepreneurs and scientists, to make a mark in this setup. So there's an organization called BIRAC, Biotechnology Ignition Council, and Women Lift Health and Gates Foundation. All these organizations are helping a lot for taking these projects to a more marketable, to a more generic ground. So yeah, so I'm really thankful to Government of India for this. And that's it. That's my presentation for today, and I'll be very happy. We'll be hiring candidates soon, and I'll be very happy to collaborate with your organization as well, and talk to your students as well if they're interested in startups. And if you have any questions, please go ahead and let me know what are your thoughts. Thank you.
Thank you very much, Dr. Anusha Aditya, for delivering such an exceptional and thought-provoking lecture. So I would like to ask the participants, if you have any question, you can raise your hand, or you can post it in the chat box. So, ma'am, as a token of our sincere appreciation and gratitude for your contribution in this FDP, we would like to present a digital memento and certificate of appreciation. May I request Dr. Anjali to deliver the memento?
Hi, Dr. Rupita. Hi, Dr. Anusha.
Hi.
So it was a great talk, fantastic talk, and we really enjoyed a lot. And I think people must have questions, but I think it's late evening, so they
I know. Just
barely there. So, and we will personally, sure, we will have some chat about your work. It was very interesting work.
Sure.
So please accept a digital memento as a token of our appreciation on behalf of our Department of Biotechnology and Bioengineering, School of Biosciences and Technology. And now I would like to close the session, and congratulations, Dr. Anusha, and thank you once again for accepting our invitation and gracing this event with your gracious presence, and for the interesting lecture.
Yes,
and
thank you.
we
Thank you,
hope
ma'am.
to collaborate in future. Thank you.
Yes.
Thank you so much for having me. Yeah.
Thank you.
So if possible, we will also sign a MOU.
Yes, yes.
Sure. Yeah.
Yes,
Sure.
yes. So I have talked to Anusha about this. We will have a discussion separately with the HOD and Dean.
Perfect. Yes. Yeah. All right.